Hello everyone, welcome back to my channel. My name is LEE ZHI XUAN. So this video is the free class video for STPM Biology Semester 2, Chapter 12, Homeostasis. So this video is special designed to those who taken STPM Biology and in Semester 2 now. So, based on the syllabus which provided from the Majlis Peperiksaan Malaysia (MPM), in this chapter there are 4 main big topics that you need to learn. So this chapter is also very long chapter, similar to the last chapter, chapter 11, reproduction, development, and growth. So, first topic is importance of homeostasis. So you not only learn about the homeostasis, but you also learn about the thermoregulation in this topic, under this topic. The next topic is the new topic that for you guys that you never learn in SPM, which is liver structure and also liver function. And the third topic is the osmoregulation is mammalian, which in human lah. So this one topic is actually you also already learned in SPM basically, including kidney structure, nephron, and urine formation as well. And then the last topic is osmoregulation in plants. So these are the 4 major topics that you will learn in this chapter.
In this lesson, I'll be using these figures to guide our explanation. If you've already purchased my notes, you'll find the full, detailed explanations written out there to follow along. If you'd like to get your own copy of these notes, feel free to contact me directly.
First, you need to know what is the meaning, the definition of the homeostasis. So actually, before you proceed this chapter, in first chapter, actually you already learn one homeostasis mechanism lah, which is the controlling of the breathing rate. Which including the chemoreceptor, carotid body, aerotic body, right? All this in breathing rate. Another mechanism that you already learned in last chapter, chapter 11, which is during the childbirth, the oxytocin hormone level, which is a positive feedback mechanism. So this is the example of the homeostasis process lah that you already learned. So this chapter actually is not the new chapter to you guys. So the definition of the homeostasis is simple like this. It is a process or mechanism of your body to maintain the constant internal environment. So in human, you need to know our internal environment is required to be constant. We cannot increase our something like body temperature. Body temperature, we cannot increase at always at high temperature rate. Also, we cannot always at the low temperature rate. We need to maintain constant. So like human temperature, normally constant is 35 to 36 degrees Celsius, something like that. So if lower than 35, it has problem. If higher than 37, you are already have also problem. So this is how process we call as homeostasis. So this is a basic mechanism. So actually similar to what you already learned in the first chapter actually. So first of all, you need to have the stimulus. Any changes in the internal environment. So in your example for first chapter is the blood carbon dioxide concentration. If your blood carbon dioxide concentration already increase, meaning that your pH of the blood is decreased, so that your receptor will be detected. So in the breathing rate, your receptor is chemoreceptor, right? In the chaotic body and iotic body. So after that, you will go through the control center, which in your brain one, inspiratory control center, inspiratory center. After that, this inspiratory center will send the impulse to your effector. So in the first breathing rate that you use as example, the effector is the lung, your lung, including the muscle, intercostal muscle, diaphragm, rib cage, all that. This is your effector. And then your effector will provide a response. Muscle either is contract, relax, or your gland either it will secrete hormone or not secrete the hormone. So this is a basic process of the homeostasis. So once it provide the response, it will change your internal environment to the return to the set point. Okay, so there are 2 mechanisms that in homeostasis Either one, positive feedback or negative feedback. So negative feedback mechanism is very common lah. Is more commonly compared to positive. Including the boosting rate that you already learned. So in this chapter, they have three main negative feedback mechanism that you need to learn lah. Thermoregulation, which is your body temperature regulation, osmoregulation, and also the blood glucose level. So this is the three main topic uh, example that you need to learn. So positive feedback mechanism, one of the example they mentioned oxytocin during the childbirth. So only one example. Yeah, only one example is for human is positive feedback mechanism one. So that's all about the positive feedback and negative feedback. So about, of course, you need to learn why homeostasis is very important. One of the important thing is to maintain the constant environment. So even though your external environment is changed significantly, meaning that maybe you are in desert. Desert normally during the day is can up to 60 degrees Celsius, but still your body internal environment, body temperature still maintain at 36 degrees Celsius. It will not change one. This is how your body to modify your body temperature. So of course your physiological activities are not instantaneous, meaning that your internal mechanism will not change by the environment immediately. If you, of course, if you stay at the desert point in 60 degrees Celsius for 2 hours, 3 hours, of course you will die because your body cannot sustain this kind of environment. But during this 3 hours, your body is able to sustain. So even though you have your external environment change dramatically, you still can survive in a short time. So this is the importance of the homeostasis. Of course, or moderate the change of internal environment only, but it will not eliminate the internal environment. And also it's enhanced by the in signal like hormone and effector to reduce the fluctuation, and then also allow the biological system to function efficiently. Less energy is wasted, and to allow the organism can survive at the wider range of habitat, meaning that human can survive at the desert place, can survive at the north point, south point, in the also very cold place also can be survived. All that lah. And also allow the enzymatic reaction lah. So if your body temperature sudden change, of course your enzyme also will be denatured, right? So homeostasis maintain the internal environment so that your enzymatic reaction still can be function as normal lah. So okay, one of the example of the thermoregulation that you need to know is thermoregulation, which the animals maintain their body temperature within a normal range. So in the real world, there are two types of the animal organism that provide different way of the thermoregulation. First one is endotherm. Another one organism is ectotherm. So endotherm is like human. All the mammalian is considered as endotherm, meaning that organism usually warm by heat generated by metabolism of the cells. But for ectotherm, this kind of organism get the heat from external sources. So meaning that in simple word, endotherm, even though the environmental temperature increase, but your internal body temperature still constant. But for this kind of organism, ectotherm, like snake, reptile, all that organism, they will change their body temperature based on the environment. If the environment is very high, their body temperature also very high. If their environment temperature is low, their body temperature is low. But for endothermic, no, their body temperature is always constant. Like human, 35 to 36 only. They will not say that even though you go at the desert point, 60 degrees Celsius, you still have your body temperature will increase up to 60 degrees Celsius. No. So in our syllabus, we only learn about endothermic. Ectothermic is not in our syllabus. You don't need to learn it. Only endothermic. So in endothermic, based on the Campbell, if you refer to the Campbell, there are 6 major ways to regulate their body temperature. First, including insulation, blood vessel contract, relax, counter current exchange, evaporation, thermogenesis, and final one, behavioral response. Okay, so we will go through it one by one. First of all is insulation. So insulation in simple word, fat, like fat. You can imagine that if the person which is very containing many fat, which obese lah, that kind of people is more able to sustain at cold environment. They will not easily to be get cold one, but they are easily to get hot. So this is what we call as insulation. So in scientific term is that insulation actually is to reduce the flow of heat between animal body and its environment. Because if the people have many high higher thickness of the fat, meaning that they can reduce the heat between animal body and environment. The heat flow is very slow. So that's why that the obese person can able to sustain at very cold environment lah, compared to the normal people. So insulation normally is found at the body surface, including hair and feather, and also the beneath layer lah, which the fat lah, our fat. So normally these two substance they're able to secrete oily substance that can able to repel the water, provide the insulating capacity of the feathers of her or fur lah to the organism. So for the organism that lack of fur, like human does not have many fur, right? So they rely on the fat lah. So for hair and feathers, normally is for animal like dog, cat, birds, all that. They are normally have many hairs and feathers. But like human like us, we more focusing on fat insulation. So this fat insulation able to transfer heat to water more rapidly than heat transfer to air. They can maintain our body core temperature without requiring more energy from food than land mammal of similar size. So it is what we call as the insulation. Insulation. So in simple word, what if you want to understand it is either through the body surface, hair and feather, or the fat lah. Okay, another one is blood vessel contract relax. So actually this one is the blood circulatory system play the role in the thermoregulation. So normally you need to know that our blood circulatory system containing majority is blood, right? Blood. So our blood component majority is plasma. So majority component of plasma is water. So you need to know water play the role in cooling effect, right? That you already learned in chapter 1. First chapter, properties, chemical properties and physical properties of water. So in response to change in the environment, animals actually can adjust their blood of amount flowing between their body core and their skin. Meaning that you need to know, even though in our surface of our skin, they also containing many blood vessels. So in response to the temperature in hot condition, meaning that hot condition, meaning that your muscle of your vessel wall in the surrounding your skin will relax, which is we call as vasodilation, to widen your blood vessels. Blood vessel. So if your blood vessel is already widened, meaning that your diameter increase, what is happening? Meaning that there are more blood will be flow to your skin surface. So blood flow in the skin, to the skin is increasing. So why they want to increase the blood flow? You need to know water, because our blood containing majority component of water. Water provide cooling effect because water has high specific heat capacity and also high specific latent of heat heat of vaporization. This is all about the revision that you already learned in Sem 1. Of course, you also need to write in the exam as well. What is happening? Not just until here, then that's all. You need to write. Of course, during cold condition, so your muscle wall will be contract, meaning that it reduce the diameter of the blood vessel. So it reduce the blood flow so that there are no less amount of the blood will be transferred to your skin. So So this is about the blood vessel contract and relax. And also another system is spatial system for counter current exchange. Okay, what is this system? Okay, so they use the goose and dolphin as example lah. Okay, so you need to know that this system actually is to allow flowing in opposite direction to reduce heat loss during cold condition. So normally this is the example of the animal that stay at the very cold environment. So especially dolphin. Because you need to know dolphins are right at the ocean, right? And inside the ocean, actually the environmental temperature is very low. It can down to 10 degrees Celsius in summer and can be negative one. It can be negative during the winter one. So this counter current exchange system is special for those animals that normally survive at very cold condition. So you need to know that in our blood we containing blood vessel, artery and vein, right? So artery and vein, what is their function? Artery is normally is transporting oxygenated blood from blood to the whole part of body. So normally it is downward. Normally it's flow downward because it flow from the heart to the body, right? To the body, right? To the foot. So similar to the dolphin, they also from blood to downward to whole part of the body. So for vein, they transfer deoxygenated blood. So they normally transfer from the down to the down like food to the body of the to the heart of the organism. So similar like dolphin, they transfer from here until to the heart. So this current exchange, this is the simple anatomy of the blood vessel, but because of this anatomy, they can provide the heat absorption between the blood capillary. Okay, what this does this mean? Meaning that artery normally is carry warm blood to the animal. Right? Because our oxygenated blood normally is warm. It actually carry the warm blood, and when they reach the food, which is very far away from the heart, your vein actually is more cooler compared to artery. Artery. So when they close contact with the vein to the opposite direction, which is the opposite direction, the heat will be transferred from arteries to the vein along the blood vessel like this. So you can see here the temperature is very high until when it reach the food, it will be very low. Why? Because the heat is already transferred to the vein, to the vein. So this can reduce the heat loss during the cold condition, meaning that your blood, you can maintain your blood temperature is equilibrium state. Will not say that artery will have higher temperature, but your vein will have low temperature. So for now, your blood is in the constant temperature. So normally near the end of the leg of the flipper, the arterial blood has been cooled to far below the animal's core temperature. The heart artery still can transfer the heat to the even colder blood in an adjacent vein. So in simple way, artery blood normally is higher than vein the temperature. So the blood in the vein will continue to absorb the heat. So the function of the vein normally is absorb your heat. It absorb the heat from the artery. On the artery. So the blood in the vein continue to absorb heat as it pass warmer, and warmer blood will traveling in the opposite direction in the artery. So this is like the what we say as counter current exchange, because the heat is counter current exchange. They exchange the heat between the artery and veins. And why this can be happen? Because the blood is flow in opposite direction. Blood for artery is flow from the heart in the center of the body to our foot, right? But for vein, they transfer the blood from the foot to the center part of the body. So as the blood in the vein exposed to the center of the body, your vein blood temperature is already something similar like the body core temperature. So this will minimize the heat loss that result from supplying blood to body parts immersed in cold water. So that in so that it maintain the vein blood temperature so that when the vein entering the core body part, your core body temperature will not be to reduce. So that it can maintain your core body temperature. Because you can imagine when you stay at the very cold condition, you imagine you are now in the ocean. It very or not say in the ocean, in the winter country, and the environment temperature is negative 10 degrees Celsius. So meaning that your vein is actually is very cold, but the outer vein is very high. If does not have this counter current exchange, your outer vein temperature also will decrease when they reach the food, but your vein will be more lower. And once your vein entering to the core body part, like the center lah, normally core body is the center, but actually your core body is have the highest temperature in the body one. Normally like 36 degrees Celsius, highest one. But due to the vein have the low temperature due to the environment, so your vein have very low temperature already. So your body core require to warm up the blood blood by transfer the heat to the vein. So they already lose the heat from the body core. So can you imagine when the body core of the temperature of the animal already reduced, can you imagine that this animal can survive at the winter condition or not? Cannot. Of course cannot. Cannot. So this heat transfer along the entire of the length of the blood vessel, maximizing the rate of heat exchange and minimizing the heat loss to the environment. So they trap the heat in the body. They don't want the heat loss from the environment one because they are already very cold. If they always release the heat to the environment, they are losing the heat. They need to maintain their heat in the body one. They cannot lose the heat anymore. So that's why this one of the importance of this system lah. So normally this system is special especially for ocean organism lah. Marine organism like dolphin lah. So this mechanism can keep the dolphin can swim in the winter environment lah. Because their muscle is always warm. And able to do the vigorous activity in the cold environment. So for the insect, it will help to maintain a high temperature in their thorax where flight muscle are located. Okay, so this is about the counter current exchange. Okay, the next one is evaporation. So evaporation actually is very easy lah. Easy lah. This meaning that when you do exercising, alright, or when you do the is very hot temperature, awak akan berpeluh kan? You are sweating. So sweating actually is the one of the way to cool down your body one. So if your environment temperature is above your body temperature, evaporation can keep your body temperature from rising by sweating. So your sweating meaning that your body is containing many water. So the evaporation of the water can keep the body temperature constant. So this is actually recap to the chapter 1 lah. Chapter 1 lah, because water have higher heat of vaporization. You want to evaporate the water is very difficult one compared to other type of the liquid, because the energy that required to evaporate one water molecule is very high. So evaporation of the sweat from the skin, water from the skin can prevent the overheating on a hot day when excess heat is generated by the sweatiness activity. So even though if you are in the hot day, you are sweaty, of course you do the exercising, sweatiness activity, vigorous activity, you also have very sweating, right? So the water will able to absorb the heat when it's evaporate, so the heat will be carried away from the skin and respiratory surface with water vapor. So this is what we call as evaporation. So another one is thermogenesis. Okay, so this one is for the endotherm. They need to maintain body temperature higher than from environment. So normally this of course is winter condition lah. The temperature is very low one, like 10 degrees Celsius, negative or even negative environment. So this kind of organism, they're able to do thermogenesis. So the endotherm can able to produce the heat to match the changing rate of the heat loss. So it is actually increased to by the muscle activity such as moving or shivering. So for the mammalian, you can for the example for the mammalian, endocrine signal can release the response to cold, cause mitochondria to increase their metabolic activity. So when you respond to call like us, when we respond to call, they will cause the mitochondria to increase their metabolic activity. So to produce more heat. So it is not to produce ATP, but it is to produce heat. So this is what we call as thermogenesis lah. Okay. Next is we want to learn about the thermoregulation in human skin. Okay. Human skin. So I think this is the anatomy structure of the human skin. So I think everyone already know it. Why? We have in human. Normally we have three major layer. The first one, the outermost one that you see on using the eyes is epidermis. After that is dermis. Dermis is the thickness part, and then the final part is the subcutaneous layer, which is the fat layer lah. They containing majority of the fat. So you can see lah. See this is the blood vessel, and then you have the sweat gland. Sweat gland to produce the sweat. Of course, containing some of the human have skin, like not skin, hair. They have the hair, something like that. So this is the anatomy structure of the skin. So in homeostasis, normally we have two situations. So in thermoregulation, of course, you have hot environment and normal environment. So when you are in very hot environment, meaning that your environmental temperature is very high, your body temperature was starting to increase. So not only in the hot environment, but when you doing exercise as well, your body temperature is increasing. So what is happening that first of all, in basic mechanism of the homeostasis, this is the stimulus. Okay, your body temperature is already changed, is stimulus. So what is detected? So you need to know your detector, your receptor. What is a receptor? So meaning that your heat loss center within the hypothalamus or thermostat is activated for cooling mechanism. Cooling mechanism. Okay, so they will send the impulse to the effector. So there are many effector. One of the effector is the blood vessel. So this will cause vasodilation. As I mentioned before, right? In hot environment, it will cause vasodilation, meaning that it increase your blood diameter. More blood will be flow from the body to the skin surface. So vasodilation will increase the transfer of body heat to the environment. So because your body inside the body is very high in temperature, it's very hot now. So you need to release the heat. So how to release the heat? This blood can release the heat when they transfer the blood from the center part of the body to the skin surface. The heat will be released through the skin surface. So this is what we call as vasodilation. So that's why that during hot environment, you need to increase the blood flow. Your blood flow will be increased to reduce the heat loss. Of course, your sweat gland also will be stimulated to secrete the sweat, so to provide the evaporation cooling effect, as I mentioned just now. So another one is the insulating layer. So in human, our insulating layer is here lah, the mist. Including the mist and also subcutaneous layer, will decrease the rate of insulation and will reduce the flow of the heat between the animal and the external environment lah. And also it will cause your hair evator muscle to relax, allowing your hair lies to the skin surface. So in hot environment, your hair evator muscle is relaxed one. So your hair is going down, that attached to the skin surface lah. So evaporation of the sweat will be reduce the body temperature, release the heat, and also, of course, you also have the behavior response. So including drinking more water, or you will bath. Bathing is one of the behavioral response. To the moving to the cooling area in another direction, which is more sustainable and minimizing the heat of exhaustion from the sun. Okay, next one is cold. When you are in very cold temperature, what is happening? So similarly, you need to know what is detected. So of course, it's also thermostat or heat gain center in the hypothalamus is stimulated, and it will send the impulse to the effector. So of course, similar effector including the blood vessel. So your blood vessel now is vasoconstriction, which will contract. You need to reduce the blood flow from the body temperature to the skin surface because they want to reduce heat loss. We increase the blood vessel diameter of blood vessel is to increase the heat loss. They want to reduce the body temperature, so they need to release out all of the heat from the body. But for now, you are in very cold environment, meaning that you need to reduce the heat. You need to minimizing the heat loss. They want to trap the heat in the body. They don't want the heat lost out from the body. So this is what we call as vasoconstriction. And of course, the skeletal muscle will rapidly contract, causing the shivering, which will generate heat. And also, your insulating layer will play the role, increase the rate of the insulation, and your hair evator muscle will be contract to pull the hair up and sweat like this. So this will increase the thickness of layer of air trap. Okay. So of course, including you can write about thermogenesis as well, as I mentioned before. Thermogenesis, which is generating of the heat by shivering through the skeletal muscle. Okay, so that's all about the cold condition in the cold environment. Okay. Environment. So that's all about the first topic. So next we will go to the second big topic, which is the liver. Okay, so I think everyone know what is liver, right? What is liver? So one of the major key function of your liver is to detoxification. Right? They detoxify all the toxin substance in the body. The one of the key major function that you already learned from primary school until now. So in STPM Biology, you not only learn about the detoxification, you also need to learn about the liver structure. So this is a liver that you see from the X-ray. When you do the X-ray screening, you see your liver is look like this lah. But when you enlarge in under microscope structure, this is the liver structure like this. So the majority that you need to know is here. This kind of structure that you need to know. Okay, so normally liver is the largest vesicle organ that locate on the right side of the body immediately beneath the diaphragm. So it normally is located at the right hand side of your body at the lower part under the diaphragm one. So and then you enlarge the liver. You can see there are many liver lobules, and each of the liver lobules they will look like this. So you can see something like this. So all of these is blood vessel here. This is blood vessel, and blood vessel all here. Okay, so we already know that liver containing three type of blood. Three type of blood vessel. Right? Hepatic portal vein, hepatic vein, and hepatic artery. Okay. So in general, hepatic artery is most easier one because it transport the oxygenated blood to the liver, from the heart to the liver. So this red color one is the hepatic artery here. Another one is vein, hepatic vein. Okay, so hepatic vein is carry deoxygenated blood from liver to vena cava. So these two blood vessels is commonly found in all of the organ in human. Of course, artery is transfer the oxygenated blood. Vein is transfer the deoxygenated blood from liver to the pump, to the heart. Sorry. Okay. Another one is portal vein, hepatic portal vein. Okay. This kind of extra blood vessel actually is connected from the small intestine. Yeah. So if you still remember that in SPM, right, nutrition topic, I think nutrition topic, the nutrition absorption and nutrient assimilation topic, you already know that any extra nutrient content like amino acid, glucose, all that will be transmitted from small intestine to liver by the hepatic portal vein. So hepatic portal vein main function is to transfer the accessing nutrient or delivering nutrient from the small intestine to the liver. So this is what of the function of the hepatic portal vein vein lah. And the basic unit of the liver is liver cell, but in scientific terms we not say liver cell, we say hepatocytes. So hepatocyte is the liver cell. It's a basic unit that contribute to the liver. And of course, in liver, they also containing the bile duct here. Bile duct. So bile duct. So bile duct is very easy one. The flow of the bile duct is opposite from the portal vein and also outer vein one. So in examination, normally they will come out this kind of structure, but they will like the arrow one. So this arrow actually is the flow direction. So if you see that the arrow which is opposite direction from here, this one is recalled as bile duct. So what is the function of bile duct? So normally bile duct is you can say like the blood vessel that transport the bile, one type of the fluid that store in the gallbladder one. So the hepatocyte also will secrete the bile, which we call as bile, bile. Secrete the bile. This is one type of the fluid, and will be transported through the bile duct to the gallbladder. So normally your bile will be stored in the gallbladder. So and this bile is important for the nutrient digestion, right? That you already learned in SPM. So where is the bile produced? It is actually produced in the liver, but they store in gallbladder. And another one, this one structure does not say. This actually is more structure that you will see in exam not like the colorful structure. So you can see here, there are many cells here we call as hepatocytes. All these cells is hepatocytes. So how to differentiate the vein, outer vein, and hepatic portal vein based on this diagram? So normally hepatic portal vein and outer vein, they are connected to each other one. They were finally combined together one. You can see here, and there were another blood vessel at the another side. One blood vessel at this side and another blood vessel at this side. So another side is vein. Of course, it's vein. So you can see here the blood is transmitted flow from here. So you can see when there are these two structure at here, you will already know one is portal vein, one is outer vein. So in labeling you can simple label lah. Label lah. Yeah, either this one is outer vein, this one is vein lah. But I think in real examination they will not come out like this lah. They will more detail lah, more detail lah. So you can see here when they have the interchange here, meaning that these two is portal vein and outer vein one. Not compulsory, not hepatic vein. If you write these two is hepatic vein, then totally wrong lah. Because the blood is flow, that interchange it at here, and then it will flow until it reach here. So this is hepatic vein at the another side. Okay, so there are another cell we call as capsule cell. Okay, this is the found in the liver to form reticular endothelial system to eliminate the bacteria one. So the major function of these capsule cell is to eliminate the bacteria and also red blood cell, all red blood cell or all erythrocyte by phagocytosis. So this is the two function. And another one is we call as sinusoid. Okay. Blood, this one, this line here is not outer V, is not vein, is not so not say that this is the not vein, not outer V. This one line which we call as sinusoid. So this is we call as sinusoid. So sinusoid actually is the pathway to allow the blood flow from the hepatic artery and hepatic portal vein into the hepatic vein. So this is the part blood flow through, and then the hepatocyte will receive all the oxygen, nutrient from this from here lah. So this is the and then another one is you see here. When you see this diagram, you can see here there are another line here, and the direction is opposite. And the major characteristic is it will not connect to the blood vessel one. Totally no. They will not connect to the blood vessel one. They are separated. So this is what we call as blood bile duct, or another bile duct is here. Bile duct is here. So this one is the line here. Line here is we call as bile canalicularis here. The bile that will be transported, produced by the hepatocytes to bile duct is we call as bile canalicularis. And at the end of the bile canalicularis is the bile duct, which is connected to the gallbladder. Your bile will be transmitted to the gallbladder and stored at there. Once they need to use is using when the nutrient digestion process. So this is a basic structure of the liver. So I hope you guys can understand what you can imagine when the exam come out, you're able to label the structure of the liver cell. Okay. Next, we go through the liver function. Okay. Liver function. So there are actually two major function in the liver. Well, first one is the carbohydrate metabolism, and another one is the protein metabolism. Okay. Okay. So in carbohydrate metabolism that you already learned in Sem 1, right? In Sem 1, you learn about glycolysis, right? Because glycolysis is happening in every cell, where you use the glucose to provide the energy. So every cell that undergoes glycolysis one. But liver not only undergoes glycolysis, they also undergoes other type of the metabolism, which is carbohydrate and protein metabolism, special for the liver. So in carbohydrate metabolism, there are three major processes that you need to learn: glycogenesis, glycogenolysis, and gluconeogenesis. So they are all of them are like the siblings. Another one is glycolysis, glycogenesis, glycogenolysis, gluconeogenesis. But glycolysis already learned in Sem 1, so of course not in this. Another one is protein metabolism, deamination, ornithine cycle, and transamination. Okay, so we need to know one by one. Okay, so first of all, carbohydrate metabolism. Okay, so this actually is controlled by two major hormones that you already learned in SPM, which is insulin and glucagon. Okay, recap to your SPM. Where you recap what you learned in SPM. For the insulin hormone, when does the insulin is secreted? We know that insulin and glucagon, both of them are produced in pancreas. And insulin is secreted from beta cell of islet of Langerhans. Glucagon is secreted from alpha cell of islet of Langerhans. This is very important. You must mention. You cannot say only pancreas. If you just write pancreas, no marks given, especially in structural question and essay question. You are required to highly specify it. Okay. In SPM, you learn insulin is secreted when the blood glucose level is very high. Glucagon, it was secreted when your blood glucose level is very low. So insulin will convert glycogen no, will convert glucose into glycogen. Glucagon will convert glycogen into glucose. Right, this one you learn in SPM, and you already learn structure of glucose and structure of glycogen. Right, glucose is monosaccharide, glycogen is polysaccharide. It is highly branched structure. They containing alpha 1,4 glycosidic bond and alpha 1,6 glycosidic bond. This one you already learned in Sem 1. So begin this topic, you are required to relate all you already learned in Sem 1 because all are related. Okay, so in insulin, your process is glycogenesis. So that's all you already learned in SPM. That's all only. About the process, metabolism, glycogenolysis, gluconeogenesis, you not learn in SPM 1. You only learn in SPM, you just only learn, insulin convert glucose into glycogen. Glucagon convert glycogen to glucose. That's all. But please, in STPM, no convert this, convert that. Convert term cannot be used in STPM. You need to use very specified. So glucose convert to glycogen. How to specify it? How to use the term? So glucose is monosaccharide. When you want to change your monosaccharide into polysaccharide, process is called condensation. So you should use condense. For glucagon, glycogen into glucose. The process is hydrolysis. So you should use hydrolyze. Hydrolyze or break down. Glycogen is break down into the glucose. Not convert that. This cannot. So in insulin, so that's all you already learned in SPM and done. This is your SPM syllabus. So in STPM syllabus, you will get more deeper. How the glucose is condensed into the glycogen. How? How the liver work? How for glucagon? How are your glucagon play the role to hydrolyze glycogen into glucose? So the condensation of the glucose into glycogen is recalled as glycogenesis. Another one is the hydrolysis of the glycogen into glucose either is glycogenolysis. Okay. Another process is gluconeogenesis. This process is the formation of the glucose by using non-carbohydrate substance. So this is extra one. The hydrolysis of glycogen into the glucose is glycogenolysis. Gluconeogenesis is not using glycogen anymore. No glycogen anymore. They use non-carbohydrate substance. Okay, so we will see it what so this is the basic metabolism blood glucose level. Glucose level. So in basically when your blood glucose level is high, your beta cell of islet of Langerhans in pancreas is stimulated, secrete the insulin so that insulin will play two major functions. One of the function is to transport it to the liver to receive the glucose and store it as glycogen. So it as glycogen. So by the glycogenesis. Glycogenesis. And at the same time, it will also go through the other part of the body so that the body cell can take up more glucose level. So it will also increase so this will decrease the blood glucose level to the set point. So the set point. So your blood glucose level will become normal. So and similarly for the low when your blood glucose level is low, your alpha cell of the islet of Langerhans in pancreas is stimulated to secrete the glucagon. So your glucagon will be transported to the liver and break down or hydrolyze the glycogen into the glucose. And this glucose will be released into the blood for other type of the cell that require glucose. So this is the process we call as glycogenolysis. So your blood glucose level will be increased to the set point and back to the normal. So this is we call as the hormone. Okay. So this is the basic principle that you already learned in SPM. Only in SPM, but in examination, you still need to like all of this. But in STPM, you will learn in deeper. What is glycogenesis? Okay, so this is the formation of glycogen from the glucose, condensation of the glucose into the glycogen. Okay, so this is the recap. What is a glycogen? So you can know that glycogen is highly bond structure, and these look like this. They containing two bond, right? Alpha 1,4, uh, alpha 1,4 glycosidic bond and alpha 1,6 glycosidic bond. Because of the formation of 1,6 bond, so they form the bond structure like this. Like this. So containing amylose and amylopectin, right? So that one you already learned in some one. So first step, you want to make glucose, only one glucose form something like that. Of course, you need to have many glucose molecule lah. So you can step one is the step one is the actually is I don't know is the first step or not glycolysis, but I already forget it. But it's still imagine that glucose is phosphorylated into glucose 6 phosphate by using hydrogenase. If not mistaken, it's the first step. First step of the glycolysis. Glucose is phosphorylated into glucose 6 phosphate by using the enzyme hydrogenase. After that, glucose 6 phosphate is isomerized into glucose 1 phosphate. They change the phosphate group from carbon number 6 to carbon number 1. So this is the second step. Second step. Second step. So I think it's clear. Okay, beginning third step, it will be complicated. Becoming complicated. Okay. They will have another chemical molecule we call as UTP. Here, ini. This is we call as UTP. Not ATP. Not ATP is called as UTP. UTP. Okay. So what is the function of UTP? UTP acts as the energy activator to activate glucose 1 phosphate first before produce glycogen. Okay. So in human, if you want to form glucose glycogen, of course, you need to activate first. Activate the glucose 1 phosphate first. So the glucose 1 phosphate will react with UTP to form UDP glucose here, this structure. Okay, so you imagine that glucose 1 phosphate, they have already 1 phosphate group. UDP, what you DP meaning that this is the triphosphate, so it containing 3 phosphate group. So it will break out 2 phosphate group from the within UTP by UDP glucose phosphorylase. Okay, so meaning that the glucose 1 phosphate, the phosphate group still remain, and then but the 2 phosphate group from the UTP will be removed. And then this 1 phosphate group will be react with the phosphate group that attached to the uridine to form the uridine diphosphate glucose, which is a UDP glucose like this. So they're containing 2 phosphate group. This part is glucose 1 phosphate 1. This part, the right hand part, is from UTP1. And another 2 phosphate group for the UTP already released out. They already released out. Okay, so this is step 3. Okay, next is step 4. So this step 4 will be repeat and repeat. Okay, repeat and repeat. Okay, what is happening here? Okay, you already have UDP glucose here, right? This is a glucose, glucose, and this is the UDP lah. So simple. UDP because it say that uridine diphosphate glucose. So glucose is here. This is a glucose, and this is called as UDP. Okay, there are another chemical component we call as glycogenin. Okay, glycogenin. So the addition of the glucose to a growing chain of the glycogen is the next step in glycogen synthesis by the glycogen synthase to form the 1,4 glycosidic bond. Okay, we know that in glycogen containing 1,4 glycosidic bond and 1,6 glycosidic bond. So first of all, you need to form 1,4 glycosidic bond first. How to form it? Okay, how form it? Using glycogen synthase here. Glycogen synthase. Here. But glycogen synthase cannot simply form a bond between 2 isolated glucose molecule. They cannot simply one. If you like this, or you want to directly use glycogen synthase to form the 1,4-glycoside bond. No such thing. Cannot. This enzyme cannot do this. This enzyme require addition step. That's why you see the more complicated step like this. Why you change to 1-phosphate, 6-phosphate, and then want to use UTP, and then another one glycogene, all that. So they need to make this complicated because the enzyme cannot simply add the bond like that. So hence the initiation of glycogen synthesis require a primer known as glycogenin here. So this is what we call as primer. This structure is act as the primer. Primer. So your UDP glucose will link with the tyrosine hydroxyl group of the glycogenin, causing release the UDP. Understand? Your UDP glucose here now, here this ni, semua ni, we add with the glycogenin. Most specified is the hydroxyl group hydroxyl group of tyrosine in glycogenin. We add with that. What is the final product? The final product will release the UDP here. It will release the UDP. And then only glucose and glucose linked with glycogenin. So only glucose linked with glycogen. UDP already reduced. No UDP anymore. UDP is already go to another way. UDP is nothing anymore. So the glycogen molecule itself acts as the catalyst for the addition of glucose until there are about 8 of them linked together. So this process will be repeated 8 times, meaning that there will another UDP glucose come here. You can see UDP glucose come here, and react with the hydroxyl group of the glucose here to form another molecule. Faham? Meaning that UDP glucose react the hydroxyl group of the glucose here, and then to form another 2 glucose molecule here. So there are 2 glucose molecule here, right? And then this will be repeat until there are 8 more glucose molecule attached here. So you can imagine that there are 1, 2, 3, 4, 5, 6, 7, 8. Akan berada dekat sini 1, 2, 3, 4, 5, 6, 7, 8. We can imagine here 1, 2, 3, 4, 5, 6, 7, 8, and then here is glycogenin. After that, glycogen synthase will take over. So this we call as primer lah. Glycogenin is as primer. So simple like the DNA polymerase that you learn in DNA synthesis, DNA replication, DNA polymerase. They require RNA primer because DNA polymerase cannot simply add the bond through the DNA. They require the primer. So simply like the glycogen synthase, they also require primer. So this primer we call as glycogenin. So first of all, already have the UDP glucose bind to the glycogenin that act as a primer. After that, they will have many UDP glucose bind to it and form at least 8 times to form that I have 8 glucose molecule attached to the glycogenin. And then after that, glycogen synthase will take over. So to form 1,4-glycoside bond. So you can imagine that the first 8 glucose molecule, you can see here, the first 8, 1, 2, 3, 4, 5, you can imagine all these, if you want to form the alpha 1,4-glycoside bond, is through the primer first. The primers first. 1, 2, 3, 4, 5, and then 6, 7, 8. These 8, you need to form the through the primer first. You require the primer called glycogenin first. After that, glycogen synthase take over. So it can form more and more glycoside bond. So that's all about the glycogenesis. So the final step is the bunching enzyme, meaning that which the there are an enzyme we call as the bunching enzyme. When they form, you can see here when they already form bunch, 1,4-glycosidic bunch as long as like this, bunching enzyme maybe it will cut down the one of the 1,4-glycosidic bond and transfer this oligosaccharide into the another carbohydrate molecule, glucose molecule here. By catalyzing the formation of 1,6-glycosidic bond. So you can imagine that when you already have very long oligosaccharide, 1,4 amylose, already have very long amylose, and after that the bunching enzyme will cut down and then transfer this cut down point oligosaccharide into the another glucose molecule to form the 1,4-glycosidic bond. So this is the overall process of glycogenesis, which is the formation of the glycogen. Okay, next is glycogenolysis. Okay, so this one is I use my handwritten one. Yeah, handwritten one, because in the book normally they just use the not use the diagram. They use the word to explain it. So I use the visual diagram to you guys. So glycogenolysis, especially as I mentioned, is the breakdown of glycogen. That function by the glucagon. So when they have the glucagon, it will break down the glycogen into glucose. So how the glycogen break down into glucose? Okay, so there are two main separate process. Okay, so glycogen is linked with the 1,4-glycosidic bond, can be cleaved by the glycogen phosphorylase through phosphorylases to form glucose 1-phosphate. So you can imagine here like this, all these, it will use the enzyme we call as glycogen phosphorylase to form glucose 1-phosphate directly, meaning that it will it will cut down, and each of them will form glucose 1-phosphate. Okay, glucose 1-phosphate. So once they already form glucose 1-phosphate, they will isomerize into glucose 6-phosphate by using phosphoglucose mutase to form glucose 6-phosphate, and then after that it will form back the glucose molecule, dephosphorylation. So similar, that's very simple. It is directly simple. Okay, complicated is here, because you need to know glycogen is branch. It is a branch structure. So branch structure meaning that branch that link with the 1,6-glycosidic bond one. You can imagine like this. Here. Okay. Dia cut. Tapi sampai satu tahap, itu enzyme ni, glycogen phosphorylase will not take over. It will take over is transferase. So because this glycogen phosphorylase only can break down the 1,4-glycosidic bond until it stops when there are 4 residue from the branch point. So you can see here, when they cut until remaining 4, this glycogen phosphorylase tak bekerja, cannot work anymore. They are stopped. It will stop. You can imagine here, potong sampai sini. 1, 1, 2, 3, 4, potong sampai sini, dia stop. Glycogen phosphorylase tak akan bekerja lah. Dia stop. Okay, stop macam mana? Dia tak akan stop punya. Dia pun kena potong juga. Macam mana potong? They will use transferase. Okay, so this transferase will break the 1,4-glycosidic bond of the limit branch, leave only one residue that link with 1,6-glycosidic bond. Meaning that this transferase, dia akan cut sini, and then pergi mana? Pergi ke sini. Dia akan cut and then form the 1,4-glycosidic bond at here. At the mother branch lah. You can imagine here, 1, 2, 3, 4, dia cut sini, sini kan, ini, and then ini 1, 2, 3 kami panggil limit branch, transfer akan cut ke sini, and then dia transfer ke sini. Ini mother branch, we say as mother branch. Okay, so how about ini, residue? This residue macam mana? So this residue will be cleaved by the alpha 1,6-glucosidase, which is a debranching enzyme to form glucose 1-phosphate. So so this enzyme similar like this lah. Because like this lah. it will cut down this glucose and release it to form glucose 1-phosphate, and then this glucose 1-phosphate will go to glucose 6-phosphate and glucose. So at the same time, they only remaining this branch. So this branch, they can be cleavage by the glycogen phosphorylase to form glucose 1-phosphate. So that's all about the glycogenolysis. Very simple. Compared to glycogenosis, it is quite simple, not hard. Okay. Next is the gluconeogenesis. Okay. So as I mentioned, gluconeogenesis is happening when glycogen is used up. You need to know that glycogen stored in the liver, but they have limit. When you are not eating at the long period, fasting at the long period, when your liver already used up all the glycogen, but your blood glucose level still very low, then gluconeogenesis will take over. They will convert all the non-carbohydrate resources to the glucose. So in simple word, one of the key process is the reversible reaction of glycolysis. Okay. So for now, you need to know glycolysis again. Although it's already summer, we were happy, yay! Already graduate, yeah. I don't want know about glycolysis anymore. No, you need to know in gluconeogenesis. Okay, so happen when glycogen is used up or during COVID cycle. So there are another term later we will see it, COVID cycle. Okay, so this is glycolysis, right? This one. If you remember that glycolysis is here, right? Sampai sini ini. Here, glucose, phosphate, ni semua kawan-kawan awak kan. Ni all your friend. You think you already forgive it? No, you cannot forgive it. Okay, so for here, I don't want to repeat all the process. So for here, I just want to focus on the some of the reaction only. The first reaction, second reaction, and the last reaction of the glycolysis. Okay, the first reaction of the glycolysis, third reaction of the glycolysis, and the final reaction of the glycolysis, actually they are irreversible. Meaning that you cannot using the same enzyme to convert glucose 6-phosphate into glucose 1. So for example, glucose want to phosphorylate into glucose 6-phosphate, you use the glucose 6-phosphatase, right? Phosphatase, right? But if you want to use you want to glucose 6-phosphate rephosphorylate into glucose, you cannot use this enzyme because it is irreversible. Similarly to here, third reaction, fructose 6-phosphate into fructose 1,6-biphosphate and also phosphoenolpyruvate into pyruvate. So as I mentioned, gluconeogenesis is the reversible of glycolysis, so they need to still want to change, want to form. So how to form? Okay, so we go through the first reaction of the gluconeogenesis, which is the final reaction of the glycolysis. Okay, so what is happening here? Apa sini? Ini apa? Okay, you can see pyruvate here, and then want to convert back into phosphoenolpyruvate, double bond. Okay, so what happening is your pyruvate here, pyruvate need to convert into oxaloacetate first. This is what we call as carboxylation. Yeah, carboxylation process, because it require carbon dioxide and require ATP. This reaction is requiring ATP. So the carboxylation process. So this is a carboxylation reaction where pyruvate is carboxylated into oxaloacetate, acetate, and require an ATP. So form oxaloacetate. After that, this oxaloacetate will be changed to phosphoenolpyruvate by PEP carboxykinase. So this is the reaction. So if this one also require GTP, and this is a process we call as the decarboxylation. Carboxylation, yeah. Right. Or decarboxylation because it release carbon dioxide. Sorry. Pyruvate is is carboxylated into oxaloacetate because it uses carbon dioxide. Oxaloacetate is decarboxylated into phosphoenolpyrate because it releases carbon dioxide. So this carbon dioxide is recycled use one. They recycle use one. So finally they form phosphoenolpyrate. And this process is happening at mitochondrial matrix one. When you want to form pyruvate into oxaloacetate, this process is formed at the mitochondrial matrix. Why? Because oxaloacetate is located in you see it at where? You see it at Krebs cycle. Krebs cycle happening at mitochondrial matrix. So it happening at mitochondrial matrix. After that, oxaloacetate is forming PP. Okay, so this is the first reaction. So first reaction. After that, you go back to the here. The I think it's the egg reaction of the gluconeogenesis. This is the egg reaction. Okay, what is happening? In egg reaction, you need to dephosphorylate photo 1,6,5-biphosphate into photo 6-phosphate, right? This is a dephosphorylation reaction. So in general, you need to use the enzyme. Okay, enzyme. So the enzyme is recalled as photo 1,6-biphosphatase. So this is a special enzyme, another enzyme, because if I'm not mistaken, the first reaction is hexokinase, right? This one is hexokinase. And then third reaction, this one enzyme is I already forget lah. But it is not the photo 1,6-biphosphatase, and another enzyme one. Another enzyme one. So this process is the dephosphorylation phosphorylation process and also hydrolysis process because it require water molecule. So it will form back to the photo 6-phosphate, and then the final reaction of the gluconeogenesis is to glucose 6-phosphate dephosphorylated into the glucose by using the glucose 6-phosphatase. It also require water molecule. So sorry, the first reaction is not glucose 6-phosphatase in glycolysis. If not mistaken, is hexokinase. Kinase enzyme, if not mistaken, is kinase one. But I already forget what kinase. Hexokinase, if not mistaken. But never mind, that one you already don't need to know. But for now, you need to know glucose 6-phosphatase. So gluconeogenesis is just like this. Actually, in examination, of course, you need to write all. If the exam come out want to describe the gluconeogenesis process, you need to write all, not just write here, write these three only. Because for now, I just want to focusing on these three. Because these three in glycolysis, they are irreversible, but you still want to come back to them. So they were using another enzyme, another reaction process. So especially here, the first reaction. You want to convert pyruvate to phosphoenolpyruvate, you need to addition one step. But for here, these two just change enzyme only. They just change enzyme. Other reaction similar that you use in glycolysis, but the reaction just reversible. So for example, you can hear 2-phosphoglycerate just isomerize into 3-phosphoglycerate by using the enzyme. I already forget. You can refer back. Similar to here. So for the ATP one, so for here, they produce ATP, right? So for produce ATP, right, in glycolysis. So for in gluconeogenesis, in reverse to form, they use ATP. So something like that. Yeah. ATP. So yeah, all about these. So that's all about the gluconeogenesis lah. So this one lah is recalled as I mentioned, this is more detailed one lah. First step is carboxylation process, and then decarboxylation, and then dephosphorylation, all that lah. Yeah. Okay. Next is Krebs cycle. Why you want to learn about gluconeogenesis? One of the key example is Krebs cycle. Okay, so what is Krebs cycle? Okay, so Krebs cycle actually is the cycle between the liver and muscle. Okay, liver and muscle. If you remember that in our muscle, when you do the vigorous exercise, you will perform anaerobic respiration, right? Right. Anaerobic respiration produce lactic acid. Okay. For now, I ask you, how this lactic acid be removed from our body? Is it remain at our body for forever? Cannot be removed? Cannot write. No, because lactic acid is acid. It is not a component or major component inside our body one. They need to be removed from our body one. So how lactic acid be removed? So there, Caloric cycle take place. So Caloric cycle is the cycling of the reaction between liver and muscle. Majority lah, liver and muscle. Okay. First of all, normally your muscle is when you're doing the vigorous exercise, your muscle will undergo anaerobic respiration, form lactic acid. Okay, your lactic acid later will accumulate in your muscle. Okay, so this lactic acid is required to be removed from our body. So gluconeogenesis will recycle this lactic acid to the glucose. So how this produced? So the lactic acid from the muscle will be transported through the blood, transported from muscle into the liver by the blood. So your lactic acid will be diffused into the blood, and then the blood will transport this lactic acid into the liver. Okay, now your liver already received the lactic acid. So your lactic acid now, lactic acid will be converted back into pyruvate using the same enzyme, reversible reaction, right? Reversible reaction, LDH enzyme. If not mistaken, is lactose dehydrogenase. It's like lactate dehydrogenase complex reaction, right? Which they will produce NADH, and to form pyruvate. After that, pyruvate will undergo gluconeogenesis to form glucose. So gluconeogenesis, ni lah, semua ni. So from the glucose, after this glucose is already formed in liver, it will diffuse into the blood, and the blood will be transported from the liver to muscle. So glucose produced in the liver, it is transported back into the skeletal muscle by the blood, where it become an energy store for the next burn of the exercise. So when you are doing exercise, this cycle will be happening rapidly because your muscle is required high amount of glucose. So your glucose high amount of glucose. So this is the main reason that athlete receive the post-event massage, and that they always cool down after the event. So they need to relax first. They cannot say that you can do the exercise or for a long time, because when you do exercise you feel sour, right? You feel like painful, sour, painful, right? Because of lactic acid. So you need to do a massage, cool down first, allow the lactic acid transported from the muscle into the liver to do all the glucogenesis process to form glucose. So this glucose can go to the skeletal muscle to repair your muscle. So you're cooling down, keep the blood flowing through the muscle, and allow the lactate and other acid to leave the muscle cell and enter the blood. So massage increase this movement from the cell to the blood. So the purpose of the massage is to increase the movement of the blood. Because you need to know muscle and liver location is still has the location, like the highway lah. You can imagine blood is like the highway, is a transport, or you want to transport lactate into the from the muscle to the liver, require time. Time. So if you do the massage, you push the blood flow more quickly, then it will more quickly reach the liver. So this is another diagram that I will show you. So normally liver is at here, and muscle normally you doing exercise, your muscle is all of your muscle is at your feet or even at here lah. But more of the example is here. Here. Yeah. So okay, so that's all about the caloric cycle. So caloric cycle is happening when you doing the vigorous exercise. So for examination, when you want to write the caloric cycle, one of the key point is when the question got mentioning about exercise. When the question mentioning about the vigorous exercise, then you should write caloric cycle. You should write. One of the point. Not write of course you can write glucagon, all that. You can write, but more focusing is caloric cycle. This is a key main point. You need to focus at here. Next, we will go through the second main function of the liver, which is protein metabolism. Okay, so there are three major processes of the protein metabolism. As I mentioned, deamination, ornithine cycle, and transamination. But deamination and ornithine cycle, they are both they are compound together lah. Okay, together lah. So deamination is the first step of the protein metabolism in the liver. So what is the deamination? So actually it is to remove the amino group of the amino acid to form ammonia, which is very toxic and soluble. Soluble. Okay. So you can see this. So you can see this amino acid, right? Glutamate, one of the example, glutamate, and then this is the what carboxyl group, carboxyl group, and this is amino group, and this is N, what we call as N, right? Basic structure of amino acid. So they have the amino group at here, so they will undergo a deamination, which will require NAD plus and water through the glutamate dehydrogenase. The majority is dehydrogenase lah. Hydrogenase to form ammonia. The key product is ammonia. Ammonium ion or ammonia also acceptable lah. This ammonia is very toxic that will be will be excreted by the kidney. Okay, so this ammonia, how this ammonia be processed? Because ammonia cannot directly excreted by the kidney one. Because I already mentioned it is very soluble. It's soluble in water, meaning that it can soluble in blood. So can you imagine in blood? Meaning that it can accumulate at any part of the organ. So ammonia need to be removed by the process we call as ornithine cycle. So this is a process of ornithine cycle. So in Sem 1, you have Krebs cycle. So in Sem 2, you have another cycle we call as ornithine cycle. Okay. So why is called ornithine cycle? Because of this. Ornithine. Okay. So what is happening in the ornithine cycle? Okay. So first of all is that condensation reaction, meaning that the ammonium ion ammonia that already formed from the deamination will react with the carbon dioxide, undergo condensation reaction that require water molecule and also 2 ATP. 2 amount of ATP to form carbamide phosphate like this. So this we call as condensation reaction. They require water and require 2 ATP. Not only one, they require 2 ATP to form carbonyl phosphate. Okay, and this is happening at the mitochondria. You can see here. This is after that, it will form the second reaction. Which the carbonyl phosphate, flammable phosphate will react with ornithine to form citrulline. So from now, they enter ornithine cycle. This first reaction still not entering ornithine cycle where this is like the link reaction lah. So carbon dioxide and ammonium ion will condense together to form carbonyl phosphate. After that, in mitochondria, the process, this first step and second step is in the mitochondria of the liver. Not all the cell in the liver, mitochondria of the liver. After that, this carbonyl phosphate will react with ornithine to form citrulline. Release one phosphate group lah. So no side product. The side product is phosphate group, but there are no adding anything. No ATP, no water, no carbon dioxide, no. They just react together to form citrulline only. So because our first substrate is ornithine in the cycle, so that's why they call ornithine cycle lah. So they form citrulline. Okay, so this citrulline will transport to cytoplasm. They will diffuse to the cytoplasm. So how they diffuse? They will diffuse through the protein we call as citrulline transporter, like protein channel lah. The specific protein channel that only allows citrulline pass through only. So the citrulline now is entering cytoplasm of the liver cell. Okay, so after that citrulline, what is next? This citrulline will react with aspartate. Aspartate here will react with aspartate to form areninosuccinate. So this process require ATP. So this is just a normal reaction. Citrulline react with aspartate to form areninosuccinate by using one ATP molecule. Okay, clear. This is step 4. Step 5. Areninosuccinate what happened is will release fumarate and to form arginine. So this is step 5. Right. Meaning that argininosuccinate will dissociate into arginine and fumarate. Okay. So fumarate, familiar with it, right? So fumarate is one of the intermediate in Krebs cycle. So this fumarate will enter Krebs cycle. They will enter into Krebs cycle. Arginine will continue to the ornithine cycle. So on arginine. Next. Okay. When you form the arginine, it will be hydrolyzed back to ornithine to form urea. And meaning that arginine is hydrolyzed into ornithine by releasing the urea. So it will release urea. So urea is the final product of the ornithine cycle, which is the waste product. So this is how urea form. So another terms of this cycle, they use urea cycle. Because urea cycle, they use the final product as the name lah, urea cycle. So urea is the final product of the ornithine cycle and it is a waste product lah, and it will be transported into the by the blood to the kidney lah for excretion process. So it is a waste. So you can imagine that the waste for the protein is urea. And this urea coming from where? Ammonia. So the ornithine, after the formation of the ornithine, will be diffused back into the mitochondria and repeat the step again and again like this. So this is a process. So the most important thing is urea. You need to know the formation of the urea. So arginine will be hydrolyzed into the ornithine by releasing urea, and this urea is waste product and will be excreted by the kidney. So this is a ornithine cycle. So easy, not too hard compared to Krebs cycle. Actually not too hard lah. Because there are actually no specified enzyme you need to remember. You just want to know the substrate product only, and the intermediate also very less. Only 1, 2, 3, 4, 5, or 5 only. Krebs cycle got 7 or 8, right? Susah. Krebs cycle susah. Ini tak susah. Ini senang. Ornithine cycle senang je. Okay. Okay, the final one is transamination. Okay, so this one they will not go through deamination and ornithine cycle. Transamination meaning that it is directly transfer amino group from one molecule to another molecule by enzyme called aminotransferase. Meaning that they transfer the amino group to another molecule. Meaning that like this, glutamate here, they're containing amino group, right? So this amino group, they will be transferred to other molecule. Transfer or amino group from one molecule to another. So they will transfer to the molecule which is containing like the amino acid, but they do not have amino group, like the alpha keto acid. Alpha keto acid, alpha keto acid, they do not containing amino group one. So they transfer this amino group into it, they will form alpha ketoglutamic acid, and forming alpha amino acid. So they forming alpha amino acid by using the key enzyme is aminotransferase. So this is the process. So by using the example, one of the example is glutamate and oxaloacetate. So when glutamate transfer the amino group into the oxaloacetate or acetate by using the enzyme glutamate aspartate amino transferase, they will form aspartate. This is another amino acid, right? Not amino acid, another molecule, aspartate. And then the glutamate will change become alpha ketoglutamate acid. Another example is alanine and alpha ketoglutamate. So alanine is amino acid, right? So when alanine transfer the amino group, it will form pyruvate. And alpha ketoglutamate, it will form glutamate. So this is another amino acid. So alanine and the enzyme we call as alanine amino transferase. So in this reaction, when you want to write it, the most important thing is here. And you can use one of the example. I recommend use this. This is a most famous example. Glutamate and the oxaloacetate one. You can use this example. Okay, so that's all about the transamination. Okay, apart from the carbohydrate and protein, this is a new syllabus, lipid metabolism, that you never learned in the old syllabus one. Right. So in old syllabus, we only learn carbohydrate and protein, right? But in new syllabus, MPM add on lipid metabolism. So of course you need to know lah. So lipid. Normally in human, our lipid is triglyceride, which is containing one glycerol and three fatty acid chains, right? So we will want to metabolize triglyceride lipid fat. How they metabolize. So lipase play the role in triglyceride hydrolysis to break down the ester bond to form glycerol and three fatty acid. So this is very simple. Lipase hydrolysis reaction to release glycerol and fatty acid. Okay, but not like that, not just like that. So you need to know glycerol metabolism. Okay, what is happening in glycerol? Okay, so glycerol containing three hydroxyl group, right? Three hydroxyl group. So what is happening here? They will go through two major pathways. Glycerol will be metabolized. The final product is 1,3-hydroxy acetone phosphate, which is the intermediate of glycolysis in the middle fifth step and sixth step one. So the key product is a 1,3-hydroxy acetone phosphate, and this is a pathway. Either the glycerol go through phosphorylation to form glycerol 3 phosphate. After that dehydrogenates under dehydrogenation under from the 1,3-hydroxy acetone phosphate. Another one is they use the dehydrogenase, dehydrogenase to form 1,3-dihydroxyl acetone, and then use ATP to form 1,3-hydroxy acetone phosphate. Either one. So this is a glycerol metabolism. Very easy. The most important thing is here. Fatty acid metabolism, fatty acid oxidation. Another term we call as beta oxidation. Okay, so what is happening here? What is happening here? Okay, fatty acid, we know that it is very long hydrocarbon chain, containing very long hydrocarbon chain. And for here, I only will explain the saturated fatty acid, which single bond one. The single bond one. For unsaturated fatty acid, I don't think MPM will come out lah. I don't think. But I don't guarantee that because I don't think MPM will come out. Because in MPM syllabus, they just say describe lipid metabolism only. So I don't think MPM come out. So I hope that MPM will not come out unsaturated one. If come out, then yeah. Because unsaturated and saturated, they are different mechanism one. But saturated is the most important because all it is a single carbon chain. So it is actually fatty acid metabolism, or we call as beta oxidation. It is actually cleavage of the hydrogen the carbon bond between the hydrocarbon at the beta carbon of the alkyl group of esterified to carb called A. Okay, very apa saya cakap. Okay, the beta carbon of the original fatty acid become the carboxyl carbon in the next stage of the degradation. The final product is acetyl CoA, then enter Krebs cycle. So I think this here. Okay, so we will go through it one by one. You can see here, this is fatty acid. Fatty acid. So at the end is carboxyl group, COOH. And there are many hydrocarbon here. Okay. First of all, they will add on CoA first. The acetyl CoA called enzyme A. Called enzyme A. So they will use ATP. Okay, so ATP. And then this coenzyme A will bind to the beta carbon. Beta carbon. And then it will react with FAD to form the FADH2. And then it will undergo hydrolysis reaction again to form back the NADH. And then finally it will form acetyl CoA. Okay, so this diagram I not very like it because very blur. Okay, we go through one by one first. First step is fatty acid activation. Okay, you need to activate your fatty acid first. Okay, what is the activate for fatty acid? Okay, this is your fatty acid. Right, this is your fatty acid. So you need to react with ATP first to form alkyl adenine intermediate. So this alkyl intermediate will react with the coenzyme A to form diester, activated alkyl group with alkyl group. So all reaction is that you are using the enzyme we call as acetyl-CoA, coenzyme A synthetase enzyme to form the diester. You need to form the diester bond here. Meaning that in the fatty acid C double bond OR, and then here is not directly linked with the coenzyme A. You need to link with the diester bond first. So this is the activation of the process. So the diester bond is formed between the carboxyl group and of the fatty acid and the thiol group of the coenzyme A here. Yeah. So this process is requiring ATP. This process is requiring ATP here. So this is the final product. Okay, you remember this? You remember this structure? Okay. After that, okay, this process, the step 1 process, is actually happening at cytoplasm. Cytoplasm 1. Okay. Beta oxidation is happening in the mitochondria. So you need to transport this intermediate diester into the mitochondria. Here, into the mitochondria. So how do you transport into mitochondria? Okay. So they will enter into the mitochondria. First, the activated fatty acid must be transported into the mitochondria so that the rest of the oxidation reaction process can be perceived. So the alkyl CoA can close the outer mitochondrial membrane but not inner membrane. Meaning that this compound diester or alkyl CoA, they use the main name or call as alkyl CoA, can pass through inner membrane. Okay, here. But they only can stay at the inner membrane. They cannot pass through the inner membrane one. So when they want to pass through inner membrane, how? So in the inner membrane, the alkyl group is transfer into the carnitine here. Carnitine. So by transesterification, and this reaction is catalyzed by the enzyme carnitine alkyl transferase to form alkyl carnitine. So meaning that alkyl group is here. Right. The alkyl group is react with the carnitine to form alkyl carnitine and release the coenzyme A. The coenzyme A is released. So this coenzyme A will return back to the cytoplasm. So this alkyl carnitine containing the fatty acid chain, the long hydrocarbon chain one, they can be able to pass through the inner membrane. Okay, once they pass through, okay, this alkyl carnitine, the alkyl group will transfer from the alkyl carnitine bind with the coenzyme A to form the alkyl CoA again. So this is just like the macam tukar kereta lah. You can imagine macam tukar kereta. Okay, dia ada satu kereta. Saya penumpang. Okay, lepas tu saya boleh directly pasu ini lubang punya. Lepas saya just masuk je. Okay. Tapi sekarang saya tak boleh masuk. Ini saya tak ada tiket lah. Macam tak ada tiket. I don't have a ticket to pass through it. So I require a ticket. So the ticket is carnitine. Okay, I need a ticket. So the ticket is carnitine. Okay, I already received the ticket. So this ticket, I will release the coenzyme A. The coenzyme A, I will release it. I will remaining the alkyl group only, which is a long hydrocarbon chain one. The C double bond, all that, and then I will add carnitine also. Okay, so I will form alkyl carnitine. Macam tumpang kereta lah. Tumpang kereta awak lah. Saya tak boleh masuk. Saya tumpang kereta. Okay, dah boleh masuk lah. Sekarang dah boleh masuk. Okay, carnitine ni dia just tumpang kereta je. Tumpang kereta. So they would still want to form ini alkyl CoA. So kereta ni carnitine will be released out, and then this alkyl group will react with the coenzyme A again to form the alkyl CoA. And then the carnitine will be released out, and this carnitine will go back to intermembrane space to fetch again another alkyl CoA in the intermembrane space. And then this alkyl CoA then will undergo beta oxidation. So this is the second step from cytoplasm to mitochondria. Okay, the next step, oxidation, beta oxidation. Now we go through beta oxidation. Okay, you already have alkyl CoA. Okay, so you can see here. This is they already label C double bond O. This one the carbon O, and then the second one and third one is alpha carbon and beta carbon here. So the cleavage is occur at beta carbon, so that's why we call as beta oxidation. Okay, so the first reaction is oxidation, meaning that fatty alkyl CoA undergo oxidation to form the alkyl CoA is oxidized to alpha beta unsaturated alkyl CoA, also called binomial CoA. The product has the trans arrangement at the double bond. This reaction is catalyzed by the FAD dependent alkyl CoA dehydrogenase here. Okay, so this is the first reaction to form reaction. Okay, so what is the purpose of the first reaction? Is actually to form double bond. So this is the oxidation reaction. And then after that, so this one is to actually to form unsaturated alkyl CoA lah. After that, this unsaturated alkyl CoA or enonial CoA or A will undergoes hydration reaction to form hydroxyl alkyl CoA. Meaning that it cleaves the double bond to become single bond, but now alpha for the alpha carbon still have H atom, but the beta carbon already has hydroxyl group. So this is hydration. And in the first process, it will release FADH2. So in one beta and the hydroxyl acyl CoA, after that it will undergo oxidation again to release an ADH to form ketoalcyl CoA. So the beta product will be beta ketoalcyl CoA. So this process will release an ADH. So this beta ketoacyl CoA, what is happening here actually is to release the hydroxyl group. Release the hydroxyl group and form double bond LA here, meaning that it actually to form the carbonyl group, the C double bond O. In glucose we have carbonyl group and ketone group, right? So actually this one is from the carbonyl group here. Okay, after that, this ketoacyl CoA will undergo cleavage reaction to to cut down the beta carbon here, to cut down the beta carbon. So it will cut down beta carbon. So another enzyme A will bind to the new hydrocarbon change. Okay, so meaning that they already cut down the carbon beta carbon. It will release the acetyl CoA. So this actually structure is acetyl CoA already here. Can you see here? After they do the oxidation process, they will form the ketone group, from the ketone group, and here C double bond all at the middle part of the hydrocarbon chain, meaning that this is already beta carbon, the hydrocarbon. So you can see here at the afterward the beta carbon, this is actually the acetyl CoA structure already. So they will cut down using the enzyme we call as thiolase to cleavage the beta carbon to release out the acetyl CoA. So there will another coenzyme A will bind to here. Another enzyme CoA will bind to here. So it will release two carbon. So you can imagine here, if there are 12 carbon, 13 carbon are including acetyl CoA, if there are 13 carbon at here, actually they will cut down here to release two carbon atom. So there will only two carbon atoms. So there will only acetyl CoA here. So this acetyl CoA will entering Krebs cycle. So the another coenzyme A will bind to here to form back fatty acid CoA, and then to do the cycle again and again until all the acetyl CoA is already formed. So this is what we call as acid beta oxidation. So so each time where beta oxidation will reduce two carbon number. So one fatty acid will not undergo only one beta oxidation. So you imagine that if one fatty acid, you can imagine if one fatty acid containing 12 carbon, 13 carbon, 14 carbon, and even 16 carbon. At the same time, they only cut down two only. So including here, 1, 2, 3, 4, and another 12. So they only cut down two. In each beta oxidation, they only cut down two carbon. So they will only release acetyl CoA, only lose out two carbon, meaning that there are still remaining 14 carbon in that fatty acid. So they will undergo another cycle, and then cut down another two, remaining 14. Another two, 12, another two, 10, 8, 6, 4, 2. The final one, 2, is directly acetyl CoA. So no coenzyme A added anymore, and this acetyl CoA will enter Krebs cycle. So this is also one of the reasons why breakdown of the fatty acid fat will release many energy. So you can imagine if one fatty acid, long fatty acid, 16 carbon, already have 8 acetyl CoA can be produced. You can imagine these 8 acetyl CoA entering Krebs cycle, and each Krebs cycle still produce ATP, NADH, FADH2. How many ATP is produced from one fatty acid chain? And you need to know each of the carbon oxidation, one cycle of beta oxidation produce one FADH2 and one NADH here. So 16 long hydrocarbon chain got already proceed 8 beta oxidation, meaning there are 8 FADH2, 8 NADH already be produced. And one acetyl CoA can produce how many FADH, NADH? 2 NADH, right? 1 FADH2, 1 GTP. So you can imagine in one 16 long hydrocarbon chain of fatty acid already can produce how many ATP? And this is not just one triglyceride. One triglyceride have 3 fatty acid. So these are all the calculation. So I don't know in real exam they will come out calculation or not. I don't know. This one I cannot promise. I cannot promise because calculation actually is university level lah. In my university time, I I got learn about this. But in STPM, I don't know got or not. Because in MPN syllabus, they just say describe lipid metabolism only. So they not request to calculate. So for this, I don't touch you lah. But if you want to know it, actually you can calculate by yourself. Right? One triglyceride, right? In examination, if you want to calculate it, they will provide you the carbon number one. Either 18, 20 carbon number, 18 carbon number, 20, 22, 24, all that. They will provide one. And then you can calculate by yourself lah. Like 20 meaning that you will undergo 10 times beta oxidation. 10 times beta oxidation will produce 10 FADH2 and 10 NADH, and you will also produce 10 acetyl-CoA. So 10 acetyl-CoA entering Krebs cycle will produce 20 NADH, 10 FADH2, 10 GTP. All that, then you multiply. One FADH2 can form 2 ATP. One NADH can form 3 ATP. Then you multiply it, then you already get the answer. So this is just a simple math question only lah. So I don't think it come out. If you got see my video, then you should able to calculate it lah. Yeah, because just a simple maths only, not very difficult one. Because the most important thing is you already know the process. You already know the process. You will release one FADH2, one NADH, then you already know all the process already. So yeah, so this is the lipid metabolism lah. Okay. Next is diabetes. This also new syllabus, which the old syllabus did not touch it. Old syllabus does not specify mention that you need to learn about diabetes. But in new syllabus, they already mentioned. Okay, so in diabetes, we know that we have two types of diabetes, type 1 and type 2. Okay, in this chapter, I will only focus on type 2 diabetes. Type 1 diabetes will be focused in next chapter. Chapter 14. Betul ke? This chapter is chapter 12, right? You're not mistaken. Not mistaken. This one is chapter 12, right? Next chapter lah, chapter 13, immunity one. Immunity. So why say that I need to separate this? Because type 1 diabetes is actually one type of the immune disease one. Actually, it's more related in immunity part. So that's not more focusing on the liver part lah. So I separate it. But I will touch it little lah. But if you want to more detail, you need to refer my next video lah. Okay, so what is diabetes? Diabetes. Okay. So diabetes, or in scientific terms, is diabetes mellitus. So it is actually caused by a deficiency of insulin or decreased response to insulin target tissue. Okay, so as you guys know that diabetes mellitus actually is one of the chronic disease in the world, right? In the world, not only in Malaysia, in the world. Yeah. So we know that if you got search some medical research for the diabetes, one of the reasons that cause diabetes is that insulin already cannot function. They still produce insulin, but they cannot function. The insulin already cannot function. So this is the causes of the diabetes. So meaning that the blood glucose level rise, but the cell are unable to take up enough glucose to meet the metabolic needs. Because when you want to make the cell to take the glucose, actually one of the key function is insulin. Insulin play the major function one. Although of course cell actually can take up the glucose naturally, but if your blood glucose level increase, your cell cannot automatically take up the glucose in large amount one. They need insulin. If no insulin, the cell just naturally take the glucose only. So instead, the fat become the main substrate for cellular respiration. So in diabetic mellitus, your blood glucose level can exceed the kidney reabsorption capacity causing the excess glucose to be excreted in the urine, which is why glycosuria is used as a diagnostic indicator. Because in diabetes patient, their blood glucose level is very high. High until the kidney cannot reabsorb all the glucose. Because although we not enter like the kidney part, but in SPM, I think you already learned it. Kidney reabsorption will absorb all the glucose molecule. You will not remain any glucose in your kidney one. But if it remain, meaning you already have diabetes. Because too many, your blood glucose too many in the blood. So there are type 1 and type 2. So type 1 is actually autoimmune disease. What is mean? Meaning that your body cannot produce insulin. Type 1 is very special. It is inherited disease, autoimmune disease, is naturally occur one. It is not the although now there are some research say that it can maybe the human-made effect, human effect, but for majority of the patient, they are the natural effect one. They are inherited. The disease is inherited. The patient cannot produce insulin and they need to always inject insulin one. This is type 1 diabetes. Okay. Another is type 2. Type 2 diabetes is very, very famous. All the majority that get diabetes is considered as type 2. Okay, so what is type 2? So it is actually metabolic disorder characterized by the insulin resistance, meaning that they can produce insulin, but the insulin cannot function. Your pancreas, your beta cells still can produce insulin, but this insulin, they cannot function. You need to know, if you still remember the last chapter, not last chapter, third chapter, control and regulation one. In control and regulation chapter, you learn about non-steroid hormone mechanism and steroid hormone mechanism. Insulin is considered as non-steroid mechanism. So, what is the non-steroid hormone mechanism? It will bind the receptor on the cell surface. So, insulin resistant meaning that either the insulin they already cannot bind to the cell, or they bind to the cell but they cannot function. Bind to the cell, they cannot function as the CAMP metabolism one. No, this is what we have in type 2 diabetes. So, in which target tissue, normally is liver, fail to respond effectively to insulin despite normal or elevated insulin level, resulting in impaired glucose uptake and persistent hyperglycemia. So, the term hyperglycemia is where the blood glucose level is very, very high in long condition. So it will develop gradually, commonly in overweight individual because your blood glucose level is very high. So of course you will overweight and involve a combination of reduced insulin sensitivity and defective insulin secretion. So when your insulin is very high, but they cannot function. So your pancreas, you already know, your pancreas will tell you, insulin got many, but why the blood glucose still cannot produce? Okay, I produce more and more insulin again. Because pancreas is like that. In homeostasis mechanism, you imagine that in homeostasis mechanism, when is the pancreas will be stimulated to secrete insulin when your blood glucose level is high? Okay, they already secrete, but the insulin cannot function. They just remain at the liver only. They just remain at there only, in the blood only. But okay, then at the same time, blood glucose level still very high. So it will stimulate again the pancreas, and pancreas will secrete more and more insulin. So it will become the bad recycle. The purpose of this actually is to require a good recycle, but due to the insulin cannot function, become the bad recycle. So this is why I say that I say that it is very dangerous lah. So this is the overall process of the type 2 diabetes. Okay, type 2 diabetes. So risk factor. So normally in type 2 diabetes can be caused by either modifiable or non-modifiable. Non-modifiable is the inherited lah. Due to your family, gene disorder, all that. This one you cannot modify one. If you got, then you got. Because you cannot identify your gene one. You cannot determine the gene. The gene is gene. You already have that gene. You cannot change any gene anymore. But modifiable. But for type 2 diabetes, this one is considered as minor one. Not many inherited is caused by type 2. But modifiable is a major risk factor for type 2. Type 2. Type 2. Modifiable is very important. Low obesity, low physical activity, and unhealthy diet. All these three is the key third important factor causing type 2 diabetes. So these two factors will cause all dysfunction, mitochondrial dysfunction, inflammation, gut dysbiosis, and also RS, which is a reactive oxygen species. So later we will see it one by one. Okay. So I just want to tell you that for those who got read my note, bought my note lah. So normally in type 2 diabetes, they are they're able to secrete the insulin, but the response of cell to insulin actually is already very less. So this will define as insulin resistant. So the insulin resistant term is coming from here. So during this state, insulin is ineffective already, and will as I say, it will increase insulin. So as I mentioned, when you insulin cannot work, but your blood glucose level still high, your pancreas still secrete many insulin. So over the time, you will cause type 2 diabetes due to your beta cell dysfunction in pancreas. Because your pancreas is over functioning, then I find up at the long time your beta cell is already overwork, and it will shut down. Tak mau kerja lah. Saya kerja setiap hari, tak boleh punya, pun masih tak ada ubah punya. Saya tak mau kerja lah, then your pancreas cell shut down. So as the degrees progress, your beta cell will change. Insulin is a secretion is unable to maintain your glucose homeostasis. So you will back a recycle. So most of the patient will obese or have a higher body fat percentage distributed predominant in the abdomen region. So this adipose tissue will itself promote insulin resistance through various inflammatory mechanisms. We call it inflammation, including increased FFA resistance and adipokine dysregulation. So lack of the physical activity also can cause the T2DM, T2DM type 2 diabetes. Okay, so in type 2 diabetes, if the person have the T2DM, what are the three major effects? First effect is defective insulin secretion by pancreatic beta cell, meaning that the target cell like liver, skeletal muscle, and adipose cell is decreased the metabolic response of insulin, meaning that the cell, your liver cell, your skeletal muscle cell already cannot respond to insulin. The insulin bind to it, but they cannot respond anymore. Another is inability insulin sensitive tissue to respond to insulin. Meaning that beta cell in the pancreas is damaged, reduce their absolute ability to secrete or release insulin. Okay, so this is another effect that your insulin maybe can still function, but your pancreas is already damaged. Your pancreas is already damaged. So they cannot produce insulin. So of course your blood glucose level also very high. Another one is inadequate compensatory insulin secretory response. So actually this one is very serious lah, where your pancreatic beta cell can no longer increase insulin output sufficiently, leading to a persistent imbalance between insulin supply and demand. So he is very already can say is very late lah. Your pancreas cell is already cannot produce insulin anymore. This one still can produce, but the amount is very less. Amount is very less. This one is already say that inadequate compensatory, meaning that your insulin cannot produce anymore. No insulin anymore. This is very serious already lah. So the most major cause are as I mentioned, obese, low physical activities, and unhealthy diet. Okay. Okay. So when you are obese, so meaning that adipose tissue is very high, right? You have high number of adipose tissue. So obesity actually is the strongest risk factor to the T2DM. So it acts as the primary trigger for a complex network for pathological change that simultaneously cause insulin resistance and beta cell failure. So it cause beta cell failure and also insulin resistance directly. Obesity. Okay, so normally in a healthy person, okay, you can see in healthy person like this one. Under insulin stimulation, your glucose uptake increase, your lipid metabolism increase, your free fatty acid will release will decrease. You will release less free fatty acid. Hypoxia decrease. Your cytokine decrease. Macrophage is decrease. All this is normal. But you will see here for the T2DM patient, in healthy patient, adipose participate in a broad range of biological process involving, among others, immunity, body weight, homeostasis, and glucose lipid metabolism. In health, insulin acts on adipose tissue in two different way. So when you have insulin at here, it either will stimulate the glucose uptake and triglyceride synthesis, or suppress the triglyceride hydrolysis and induce the uptake of the free fatty acid and glycerol from circulation. So adipose tissue actually for insulin is very good one. So, however, in the patient that have many adipose tissue, your adipose tissue is very large compared to normal one. Your adipose tissue is very large, a state known as hypertrophy. So this hypertrophy cell outgo their oxygen apply, leading to hypoxia and the activation of the transcription factor hypoxia inducible factor. So actually it is trigger inflammation. You already trigger inflammation. So the cellular stress trigger the pro-inflammatory cytokine. You can see here, anti-inflammatory cytokine, pro-inflammatory macrophage. If these two are increasing, meaning that you are actually causing inflammation. You're causing inflammation to your adipose tissue. Okay, so when you're causing inflammation, how is happening is that your nutrient is overload, causing a toxic build-up of lipid intermediate, like dialcyl glycerol (DAG), and another term is we call as ceramide (ECER). So these two actually is very toxic intermediate when inflammation is occur. So this what is happening when these two DAG and CER occur. So it will occur. So this final result will cause the insulin resistance. The two product what I mentioned, the dialcyl glycerol DAG, and another one is ceramide CER. The final result is that they will undergo the series of the metabolism, but I don't think the syllabus need to know all that metabolism, but I just give you a final result is that your adipose tissue become resistant to the insulin. Because due to that two product, the alcyl glycerol and ceramide, DAG and CER, they will causing insulin resistance. So meaning that your adipose tissue is able to accept insulin already. They cannot respond to the insulin already. Meaning that they cannot absorb glucose and suppress the breakdown of fat, which is a massive constitutive leak of free fatty acid into the bloodstream that eventually damage other organs like the liver and muscle. So, in general, normal tissue in healthy adipose tissue, when insulin is coming, glucose uptake, it will increase the triglyceride synthesis, and suppress triglyceride hydrolysis. It will not release more fatty acid, free fatty acid. free fatty acid, and uptake of the free fatty acid and glycerol for the circulation. So it maintain the lipid metabolism is very stable. But when you are in a type 2, why say that obese patient easily to get type 2? Because your adipose tissue is very large, big and large. Big and large already. So it will cause accumulation of the inflammation process, resulting in the lipid intermediate like DAG and CER, and DAG and CER causing insulin resistance. So when the insulin is resistant, meaning that your free fatty acid release, triglyceride synthesis decrease, and cannot accept any glucose anymore. So this is how the effect of the adipose tissue lead to T2DM. Okay. Of course, effect to the liver as well. Okay. So effect to the liver. So excess free fatty acid, as I mentioned, this one, when you have many free fatty acid released from the dysfunctional adipose tissue, when it travel to the liver, and they will store as the triglyceride, causing fatty liver. Fatty liver. And the build up of the free fatty acid will reduce the insulin signal by disrupting the pathway in the hepatocyte. Meaning that if you already have, if you causing the fatty liver, your insulin cannot bind to the hepatocytes anymore. Cannot bind to the hepatocyte. It will affect the pathway along the hepatocyte. So the pathway that you already learned is CMP, right? CMP pathway. So disrupt the CMP pathway, then the hepatocyte cannot respond to the insulin. Then of course, no, all that insulin is glycogenesis, right? No glycogenesis process happen. So this is effect to the liver, and because the liver is no longer to respond to the signal, so it will continue gluconeogenesis. Even blood glucose level is already high, which will cause more serious hyperglycemia. Gluconeogenesis, because insulin is able to inhibit gluconeogenesis one, but due to your insulin cannot function, but your glucagon still function. But so it will make the situation become more worse. Of course, effect to your pancreas. Okay, what is the effect? When you have the high blood sugar or high free fatty acid, create a toxic environment that physically damage the beta cell. So you can see it is a bad cycle. Only caused by adipose tissue only. The free fatty acid. The free fatty acid is very key. Can say is a key toxic compound to all of us. So if damage the beta cell, high demand of insulin will cause the ER stress, which is pancreas ER, and generation of the reactive oxygen species, which will inhibit the calcium signal needed for the insulin to release. So will activate the proapoptonic signal and induce local inflammation and leading to beta cell death and reduce insulin production. So meaning that actually this one is say that when you are heavy many FFA, you will cause the toxic environment, so damage the beta cell. So the beta cell, and it will also generate reactive oxygen species, which will inhibit the calcium signal needed for insulin to release. So you want to make the beta cell to release insulin, they will require calcium signal lah. But this calcium signal already inhibited by the ROS. So if you do it like this, it will induce local inflammation, causing your beta cell die. Once your beta cell die, insulin cannot be produced anymore. So it is actually you will make a recycle lah. Cycle and cycle, bad cycle, but it is a bad cycle. Okay. So next is how low physical activities cause type 2 diabetes. Okay. So when you do a sedentary lifestyle, sedentary meaning that you only sit at there, you're not moving anymore, just sit at the computer, play game, all that, play game, all that. you will trigger a metabolic perfect storm that link to the obesity to the type 2 diabetes by disrupting the glucose regulation across multiple system. Multiple system. So, physically, when you in skeletal muscle, normally in our muscle, muscle actually is very important for the pancreatic tissue to your muscle that can able to use the glucose. But for now, because you still away sit down at there, your muscle is not moving, not working. So your muscle also don't need to use the glucose. Tak perlu guna glucose lah. Don't need to use the glucose. So your muscle contraction is reduced. So diminish the blood flow and impair the insulin-independent glucose uptake. So, inactivity will prevent the translocation of the glucose transporter GLTU4 here to the cell membrane, causing the glucose to remain in the bloodstream and resulting in hyperglycemia. So, meaning that when you are not activate, you are in sedentary lifestyle, your GLUT4, this is the one of the glucose transporter in the skeletal muscle will be inactivate. Because your muscle already don't need work. Because you just always sit down at there or barling at there or you sit on the bed or no exercising, you just play a game only, not walking, no jogging, no exercising, your skeletal muscle cannot contract. So your skeletal muscle don't need use glucose. So they already automatically know, ini orang tak perlu glucose punya. So saya tak perlu GLUT4 lah. The glucose reporter saya tak perlu lah. So tak perlu, meaning that it will cause the hyperglycemia. Meaning that when in the longer time, at the long period, all the blood glucose is stay at the blood. They will not be able to absorb by the skeletal muscle anymore. Your muscle cannot absorb anymore because your glucose transporter already reduced, already inactivate, cannot be anymore. So this will cause hyperglycemia. So at the same time, physical inactivity promote accumulation of fat. Promote fat. Well, it will cause inflammation. You see the pro-inflammatory cytokine like TNF alpha and interleukin 1 beta. All this is causing inflammation. It will cause inflammation. Inflammation. So if you cause inflammation, meaning that it will cause the insulin resistance through adipose tissue dysfunction and the release of the pro-inflammatory signal. So this state is further characterized by the elevated level of the inflammatory mediatory, the latter of which directly impair pancreatic beta cell function and promote beta cell homeostasis, reducing insulin secretion. So that one is more focusing on the pancreas part lah. And then finally is the environment is further compromised by the loss of protective mytokine and antioxidant, creating a chronic inflammation, oxidative stress that ultimately break down the body's ability to manage blood sugar. Meaning that we will still like this, including myokine like the irisin that enhance glucocorticoids and endogenous antioxidants such as glutathione GAH, will resulting in increased oxidative stress that damage insulin sensitive tissue and further causing serious. So this is how low physical activity causing T2DM. Okay, so the next one is the healthy diet. Healthy diet also very important. So there are two major parts: chronic nutrient overload and oxidative stress. Okay, when you eat too much, eating too much, what is happening? Okay, so high calorie diet, like fast food, or you eat too much buffet. Buffet bagus tak? Fast food, makan junk food, all that, KFC, McDonald's, all that, very good. But what will happen? You will see, will induce metabolic dysfunction, oversupply the mitochondria with electron, leading to inefficient nutrient oxygen and reduced ATP to oxygen ratio. So, meaning that actually when you eat high calorie diet, actually you already increase your blood glucose level at the very short time. So that your mitochondria actually flooded with the excessive supply of the electron from the oxidation of this nutrient. So this metabolite will bottleneck result in the accumulation of the toxic lipid intermediates such as DAG and CR, as I mentioned before. Alongside the leakage of the electron from the electron transport chain. Okay, electron transport chain. So your cell actually already cannot process the excess nutrient efficiently. So it will begin to accumulate these two kind of toxin already. Okay, so when it can accumulate this kind of toxin, your electron transport chain actually is unable to handle the excessive electron input from the nutrient overload. So your electrons starting to leak out and transfer directly to the oxygen. Only to the oxygen to generate reactive oxygen species. So including superoxide and hydrogen peroxide, which will damage your mitochondrial DNA, protein, and membrane. RS is very, very dangerous one. Normally, what we heard about RS normally is UV light. When you go to the sunburning process. But for now, RS is very general when you eat high calorie diet. One of the major. So when you this combine of the presence of RS and lipid toxin, these two will inhibit critical signaling like RS1 AKT effectively, dismantling the insulin signaling pathway. So in general, if you eat too much high calorie diet, you already accumulate toxic lipid toxin called DAG and CER. At the same time, you also causing the leakage of the electron from ETC because oxidation of your food will release many electrons. So this electron cannot be handled by ETC. Too much. ETC have limit. So the liquid electron will causing react with oxygen to form RS lah. And this RS and these two RS and intermediate lipid intermediate will affect the insulin signaling pathway, meaning that the insulin bind to the liver cell, bind to the skeletal muscle, or bind to the adipose tissue, they cannot signal your T cell to work anymore. Okay. Another one is glucolipotoxicity and beta cell failure. Okay. This what happened is that when you didn't eat too much, it will trigger a state of glucolipotoxicity that places the pancreatic beta cell under fatal metabolic pressure. So meaning that your beta cell actually is at the toxic environment lah. So you will release many free fatty acid and force your pancreas work beyond its capacity and activating the unfolded reporting response and disrupting the calcium signaling pathway necessary for insulin secretion. So as I mentioned before, for pancreas, want to secrete insulin, actually they have a calcium signaling pathway. Calcium is very important. But due to the FFA, you already disrupt the calcium signaling pathway. So yeah, you cannot secrete the insulin lah. So as the demand for insulin spike, the cell will begin to accumulate this misfolded protein that can poison the cellular environment. So sustain meaning that when you are meaning that the insulin pancreas not able to produce the insulin but produce other type of protein. They already produce other type of protein already. When these accumulate to each other, will cause the toxic environment. And this toxic state persists, your beta cell transition from functional exfunctional to programmed cell death will resulting in a permanent reduction of beta cell mass. So your body system also know one. When you this beta cell should be able to produce insulin, but they produce other type of protein. So other type of protein, meaning that this beta cell already got problem. So they need to destroy this beta cell, prevent this fake beta cell produce the toxic environment already. But beta cell, when it destroy, you want to recover back, not easy. You destroy the beta cell, then no insulin already. So your beta cell mass decrease. So terminal decline in insulin production capacity represent the foundation of the T2DM, as the pancreas can no longer compensate for systemic insulin resistance already. So this meaning that meaning that cannot long produce enough insulin to cover the insulin resistance. Because you need to know that insulin resistance at the beginning part, the insulin still can function, but you need to very high concentration. But if you require too high concentration, and based on this free fatty acid, intermediate toxic, amino toxic, and excess of glucose in the blood, you will cause beta cell cannot function anymore. So it is a very bad cycle. Because your cell now require high amount of insulin. For example, in normal situation, your skeletal muscle actually just require 5 ml, 5 gram per liter of the insulin already enough. But for a patient, they require 50 gram per liter. But your pancreas still can produce, But if due to your free fatty acid is too high, your glucose is too high, already cause glucose depotoxicity to your pancreas, to your beta cell. So your beta cell is already destroyed, they cannot produce glucose. So you will go back the very bad recycle. And due to no insulin anymore, if there are no insulin anymore in your blood, in your body, then unfortunately, you will get the D2DM. So yeah, this is a mechanism about the how the beta cell produce insulin. So this is a very general one. This is what we call as calcium ion. Now, calcium ion is very important for CAMP pathway one. CAMP pathway. We can see insulin release is primarily triggered by a response to a high concentration, right? Through the GLU transporter, meaning the glucose is transported here into the beta cell, and glucose beta cell, the latter will enable calcium ion influx, triggering insulin exocytosis, and this will channel like the contribute to calcium ion mobilization and insulin secretion. So calcium is very important. But due to when you have free fatty acid, you break down the calcium ion. You break down here. It's this. Calcium ion cannot enter into the cell. So when the calcium ion cannot enter into the cell, then no insulin. You already destroy the beta. So these are you can see here. Calcium ion leading to ROA generation, reactive oxygen species, inhibit calcium ion mobilization, and activate post-aspartic signal. Aspartic signal. So your calcium ion cannot do anything already. Cannot do anything already. So no nothing. Nothing. So this is very dangerous. Your glucose is increased, but no insulin can be produced. Your insulin cannot be produced because calcium ion able to let your insulin to be produced one. But for now, your calcium ion cannot function anymore due to ROS and also other type lah. So that's all about the how the healthy diet destroy your beta cell. So actually they are related lah. How unhealthy diet, obese, and the sedentary lifestyle, no low physical activities, all that actually they are related. The most important thing is calcium ion, ROS, hyperglycemia, all that. Yeah. So about the treatment of T2DM, okay, so this is the general treatment that you can normally we can do. Weight loss. Because majority of the type 2 DM person, diabetic person, they are overweight. So you of course you need to do a weight loss. So weight loss. And then the other one is carbohydrate, the nutrient intake. All the nutrient intake you need to control. Carbohydrate intake, you should control it. Don't drink the carbohydrate drink, Sasi, Coca-Cola, all that, gas drink cannot eat, high sugar drink, all that you should avoid. And avoid sugar intake, of course, fat intake, fiber intake, you should consume more. High density of fiber will improve the glycemic control and cardiovascular risk factor. alcohol, sodium, fat. This also you need to decrease. And exercising, very important. Exercise is very important. You should do exercise more. So that's all. But for the medical one, this is a medical one lah. Simple name lah. This is all the uh scientific name. Metformin, sulfonylureas, malitate, alpha glucosidase inhibitor, dialgenonin, sodium glucose co-transporter 2 inhibitor. This is a normal one. Normal one. If you actually go to Vision, go to pharmacy book, actually more than that lah. But this is just a normal one, normal one. And also treatment or this one is oral one, you eat one. Also you got inject one, so inject you can inject glucagon like peptide 1 receptor agonist and insulin therapy lah. Insulin therapy. So normally I explain about the oral one. I use one example to explain about so you can more in detail. So for metformin, normally actually it is to deliver to the liver from the intestine and to inhibit gluconeogenesis one. Gluconeogenesis. So actually this one to decrease the fasting blood glucose lah. So as I mentioned in type 2 diabetes, gluconeogenesis still happen. So this one actually to inhibit gluconeogenesis. So that even in fasting period, your blood glucose will not so high. So high. Because why gluconeogenesis happening is that during fasting period. During fasting period. Because you are hungry and then the blood glucose and maybe is not enough. Glucagon is secreted. So yeah, so this is a metformin. And another one I can say is another is this lah. Diazolidinenoin. Very difficult. So actually this one is to increase insulin sensitivity lah. Acting on the muscle, adipose tissue, and liver to increase the glucose utilization and decrease glucose production. So this one is to make the cell, the effector cell, like liver cell, to increase the effectiveness of the glucose so that it can respond to the insulin. So it can improve your blood glucose level by preserving the pancreatic beta cell function. Okay. Next is injection. For insulin therapy, I think I don't need to talk much, right? It's just like really use insulin. Supplement, or replace the insufficient body insulin to regulate the blood glucose. You directly inject insulin into your body. This one is the new one, glucagon like peptide 1 receptor agonist. This one actually is to stimulate insulin release in a glucose-dependent manner. Meaning that it does not require glucose one. It was automatically secrete insulin even though the blood glucose level is still low. So it will decrease the secretion of the glucagon. So slow the gastric emptying. So that's all about the treatment of the type 2 diabetes. So this is just an overview of the type 1 diabetes. So as I mentioned, this is the autoimmune disease where the body cannot produce insulin. Your beta cell is already destroyed, so that it will lack of insulin, your glucagon increase, so it will cause all this, no insulin at all. So this is just overview only. We will talk about more detail in next chapter 13, not chapter 14. Next, we will go to the osmoregulation in animal, which is the third big topic of this chapter. So first of all, you need to know about the structure of the mammalian kidney, which is the nephron structure. So this one I just go through only because I think you already learned in SPM, right? So you know that kidney is the bean-shaped structure, normally allocated at the back of the abdominal cavity of the thoracic and lumbar vertebra at there. Okay. So normally it's just like the bean-shaped structure, right? Dark red color bean-shaped structure, and then we have two blood vessels, which vena artery and vena vena. So artery, of course, is transport oxygenated blood, and vein is transport deoxygenated blood. So for artery, we have afferent and efferent one. Efferent and efferent, and then the thickness of the efferent and efferent is efferent is more thicker than efferent. Yeah. So this one I think you all already know that. So the UV will produce first will be stored in the vena pelvis, and then the UV will exit through a dark core elevator. So all about that. So yeah, so I think nothing much. Yeah. And then presomal tubule, right? The first presomal tubule, and then distal tubule collecting loop of hinder at here, and then what else that you need to know? Here lah. Bosman capsule and glomerulus. Maybe some of you are confusing lah. Bosman capsule actually is the kidney part one. Actually, it is in the kidney part, under kidney part. But glomerulus is the blood capillaries. So when they point out the blood capillaries part is glomerulus. Bosman capsule is at the kidney part one. This is what we call as Bosman capsule. So hopefully you guys can able to uh differentiate it lah. Okay. So I think that's all about the kidney structure. Nothing. Okay. Urine formation. Okay. So this urine formation similar to the SPM actually. There are only three processes: ultrafiltration, reabsorption, and also secretion. So ultrafiltration open at the glomerulus and Bosman capsule, and reabsorption is happening at tubule, including your loop of hinder, convoluted tubule, and distal tubule. Distal tubule lah. But secretion happening at distal tubule and collecting duct lah. Mostly at collecting duct. Okay. So ultrafiltration. Okay, so this is ultrafiltration where it is a filter, because I already say filter, right? So it is actually a passive process that high hydrostatic pressure force the small molecule out of the blood plasma at the glomerulus and enter into the Bosman capsule here. So this is a afferent neuron, right? Afferent neuron pass through due to the high hydrostatic pressure. It will force out all the small molecule, including the blood, including the any nutrient like the salt, glucose, amino acid, all that they will force out into go into the Bosman not Bosman capsule both into the Bosman capsule from the glomerular capillaries. So this is a more detailed structure. So I just want to tell you that mention about that glomerulus capillaries. Here you can see here glomerulus capillary, when they enlarge, it look like this one, something like this, or you can see this, something like this. Anything like this one. So this is afferent neuron, efferent neuron. Blood come from here, come from here, and then come from here, and then it will come back from here. Okay, so glomerular capillary are the network of the capillary from the afferent neuron and rejoining to form more thinner efferent neuron. So afferent neuron is more thicker than efferent neuron. So everyone already know that, right? So the capillaries wall in the glomerulus are made up of layer of endothelial cell which containing many pores. So you can see here, this is the glomerular capillaries, and it is covered by the endothelial cells one. Endothelial cell one. And then in the endothelial cell, there are many pores at there. Awak boleh tengok ada banyak lubang dekat sana. Dekat sana. And the pore is very large. Very large one. So therefore, that is the large one. So due to this pore is very large, that lubang dia sangat besar, actually you can, even the red blood cell can pass through. So they don't want the red blood cell to pass through. They are the membrane cover the endothelial cell called glomerular basement membrane. Glomerular basement membrane, like this. So membrane that are made from the mucopolysaccharide protein and collagen fiber. So you can see these. This is a lumen capillary, and then this is endothelial cell, and this is the the membrane. This is the membrane. So it separates the glomerulus from the capsule, and it also prevent the blood cell, platelet, and plasma molecule enter the Boltzmann capsule. Okay, Boltzmann capsule. So why filtration? Why filtration is happening? One of the key reasons is that high hydrostatic pressure. High hydrostatic pressure of the afferent arteriole due to the high resistance to outflow, which is smaller in diameter than the afferent neuron. Neuron. So high hydrostatic pressure force out many blood component out from the glomerulus into the Boltzmann capsule, including amino acid, glucose, water, of course water, and also salt, and also other the excretory product and urea, all that. And then the highly coiled glomerulus capillary also provide larger surface area to facilitate the efficient high volume filtration of blood plasma into the glomerular filtrate. And also the glomerular capillaries contain numerous small pore that between the endothelial cell that form their wall, make the glomerular capillary more porous than typical capillaries. So you can see this diagram. This diagram. Both glomerular capillary is covered by the endothelial cell, which containing many pores, like this one, and then after that, it will containing many membrane like this. So the wall of the Boltzmann capsule is contact with the capillary consists of the spacious epithelial cell called pedocyte here. Pedocyte. So these cells have the numerous cytoplasmic extension called foot process that cover the glomerular basement membrane. So you can see this lah. Yeah, pedocyte. This is what you call as foot process. Dia ada banyak-banyak cabang-cabang dekat sini lah. Ini kami panggil food process lah. So food process of the adjacent pedocyte are separated by narrow gap called filtration slit. Here. Ini. Food process of pedocyte, and then this is what we call as filtration slit. So the porous wall of the glomerulus capillary and the filtration slit of the pedocyte form a filtration membrane that permit the fluid and small solute dissolve into the plasma. So you can imagine that this is a food process of the pedocyte, and then this is a filtration slit, so that it can able to component like glucose, amino acid, sodium, potassium, and urea, salt, all that can be passed through from here. So similar like this this is a filtration slit filtration slit, and then this is a podocyte. This one is a podocyte. This is an endothelial cell, the membrane, endothelial cell, and then this is a GPM, and then this is a pedocyte. So there are many layers. The capillaries is covered by the endothelial layer first, endothelial layer here. Endothelial layer with many pores, capillary pores. After that, it is covered by the membrane. We call as the cover the membrane, cover the endothelial cell called glomerular basement membrane. GBM here. This green color one. So this glomerular basement membrane is contact with the Boltzmann capsule. This is actually is the region for the Boltzmann capsule. That contact with the capillaries consists of a epithelial cell called pedocyte. Pedocyte here. This is called a pedocyte. So there are in the pedocyte there are many food process. So you can see this blue color one is many pedocyte here. Food process of pedocyte. And then here is the filtration slit. So your blood component is passed through from here into here, from here into here, from here into here. So that's all about the ultrafiltration process, how the ultrafiltration process occur. Okay, next is about the reabsorption. So normally reabsorption is to absorb all the useful material in the proximal tubule proximal tubule part, and then in the loop of hinder normally is water and salt, and distal tubule is focusing on water and salt as well lah. So even in distal convoluted tubule also got reabsorption, but reabsorption occur mostly we are focusing on this part lah. These three part. Okay, three part. Okay, so when you talk about proximal tubule, so normally majority of the component in component like glucose, all the useful component, glucose, amino acid, salts, majority are reabsorbed at here. Meaning that in loop or hinder, they should no glucose, no amino acid, no salt, not say salt, amino acid, no potassium at all. Especially glucose and amino acid because they are useful. So they should not uh, uh, present at here anymore, at the loop or hinder distal tubule anymore. Because majority are already reabsorbed at proximal tubule. Okay. Proximal tubule. Okay, so what is happening in proximal tubule? So this is a tubule, right? This is a proximal tubule, and this is the proximal tubule, and this is the epithelial cells. Epithelial cell. Epithelial cell of the proximal tubule, and this is the interstitial space, and this is a blood capillary in the kidney. So you will refer to the structure of the kidney. Normally is the peritubular capillaries at here. Peritubular capillary they call as. Okay. Peritubular capillaries. So at proximal tubule, the tubule is lined with a single layer of the cubular epithelial cell with extensive microvillus of plasma membrane. Like here. They are epithelial cells that cover the proximal tubule one. So the outer membrane of the epithelial cell are covered with base membrane and is inadjuvated to form basal channels. So the transfer of the positive charge of the tubule drive the passive transport of the chloride ion or chloride ion. So water is diffused to the epithelial cell and then to interstitial fluid by osmosis process. So got water, amino acid, glucose, potassium ions, and sodium potassium ion pump also can be occur at here, and glucose, amino acid, potassium ion, vitamin, and other essential will actively or passively transport to the interstitial fluid and then into the peritubular capillaries. So either active or passive but majority is passive transport Majority is passive transport. The active transport normally is sodium potassium pump. So beside the waste product like urea also can be passed through by the proximal tubule will be absorbed by the proximal tubule. So the reabsorption of water generate the concentration gradient between the filtrates and interstitial fluid. So this will initiate the reabsorption of the urea by diffusion. So all about in simple terms, in proximal tubule, all of the sodium, glucose, vitamin, amino acid, water will be reabsorbed into the epithelial cell first, and then it will go through the interstitial space, and then finally go through the capillary. So that's all about the reabsorption in proximal tubule. Okay, so another part is very important thing is reabsorption in loop of hinder and distal convoluted tubule. So we were more focusing on loop of hinder first. So now is at here. Here. So you can see the diagram. The blue color is passive transport. The red color is active transport. So you can see here in the descending loop of hinder is more thinner compared to the ascending loop. In ascending loop, it is more thicker compared to the descending part. Descending part of on the thin ascending part. Okay, so once the filtrate already entering the descending part of the loop of hinder, only water is absorbed. Salt is not absorbed. Only water is absorbed. So the large because in the tubule they have large amount of water channel formed by aquaporin protein. So more water is absorbed, so there is no channel for salt. So other molecules resulting in epithelial cell are not permeable. So it is only permeable to water only. So this means that water has to be diffused out from the descending loop of hinder by osmosis. Because only water, right? All the water. Thus, the interstitial fluid must be hyperosmotic to the filtrate, meaning that because it is a passive transport, meaning that the outer part should be very high concentrate lah, so that the water can be diffused out from the loop or hinder into the interstitial space at here. Okay, so the condition is maintained along the entire length of the descending lead because the osmolarity of the interstitial fluid increase from the cortex to the medulla. Because you need to know that in the medulla structure, they have cortex and medulla, right? In cortex, normally, it is very dilute. Interstitial space. I am talking about interstitial space. Cortex is very dilute one because many water is diffusing out, especially here. In the principal tubule and also distal tubule. But when they entering the medulla, especially in the inner region, the concentration of the solute is very, very high compared to the upper one. Meaning that water is easily to be diffused out when they come along the descending part of the loop or handle. Meaning that at here, many water will be reabsorbed. So as a result, the filter loads the water and increase its solute concentration along the journey down the descending loop. So meaning that your filtrate will become very highly concentrated when they go along the descending part of the loop or hinder. Next, we will go through the ascending part. Okay, so ascending part, it will form two different parts. The first part is more thin part. And then after that, you will see is thicker part. But the similarly is they are absorb salt. They only absorb salt. They will not absorb water. Meaning that in ascending part, it is more permeable to salt rather than water. So because it does not have any water channel, but now it has salt channel. So it is more permeable to the salt, which NaCl lah. So in thin segment, highly concentrated of the filtrate NaCl will diffuse out the interstitial fluid by diffusion. So this movement of NaCl maintain high osmolarity of the interstitial fluid in the medulla region. So it is actually like the balance lah. You can imagine that at here, in natural condition, medulla region need to very highly concentrate compared to outer and cortex. So when you want to maintain the very highly concentrate region, when your filtrate is coming out, water is already diffused out. Once the water is already at this region, meaning that it will become less concentrate because already got water. So when you enter at here, water already diffused out, meaning that here is already less concentrate. So you need to maintain the concentrate region. Now, NaCl need to be diffused out so that it can maintain the high osmolarity in the interstitial fluid of the medulla region. But still not enough because NaCl is passive diffusion. After when go through up here, the salt concentration for the filtrate and the medulla region actually is this one is lower than the filtrate is lower than the outer medulla one. Because here is already in the region that water is higher compared to inner region. But you still need to maintain the thick osmolarity region, so you still need to absorb NaCl, meaning that you need require active transport. So your active transport is at the upper region. At the thick what we say that the thick segment it is more thicker part. So that more NaCl is absorbed, so it maintains a high salt concentration in the interior of the kidney and enable kidney to form concentrated urine. So such counter current system will expend energy to create a concentration gradient are also called as counter current multiplier system. Similarly, like the as I mentioned in thermoregulation. So this filter is actually hypoosmotic to body fluid by the time as it reach the tubule. Once they already all the NaCl is already active transported, now the filtrate is hypoosmotic, meaning water is more than salt already. And in the distal tubule, it will play major role in regulating potassium ion and sodium chloride ion, sodium ion concentration of the body fluid lah. So meaning that this regulation involves the potassium ion secreted into the filtrate and NaCl we absorb from the filtrate. So it is active transport process. Because at now you need to know that transport process. And this so this is a process of the distal tubule. So in distal tubule, NaCl and potassium are regulated, and water also will be reabsorbed as well. That's right. Okay. Next is about secretion. So secretion is actually not too much. So actually it's an expulsion of unwanted substance and excess ion are secreted into the filtrate before it's secreted as urine. So normally in proxima, some material are actively secreted into filtrate from the interstitial fluid. Also, for example, like drug and alcohol that have been processed in the liver will diffuse from the petrotubular capillary into interstitial fluid. They were actively secreted into the lumen or proxima. In the distal cavity tubule, the potassium ion and hydrogen ion are secreted into the filtrate for the interstitial fluid for regulation. And lastly, this substance will be eliminated from the body as the urine. Okay. Okay. Next is about the another type of nephron, which is juxtamedullary nephron in concentrated urine formation. Okay, if you want to form concentrated urine in the juxtamedullary nephron, it is actually require the uh relationship between the absorption of the water and NaCl. So as I mentioned before, the cortex is osmolarity is very low, but in the inner region, your osmolarity is very high. So that it can produce high hyper uh concentrated urine here. Because you imagine if the inner part is very low low in osmolarity, meaning that when the filtrate enter until here, the all the waste product will be reabsorbed here. You can imagine all the waste product will be reabsorbed here, because already not maintained at the absorb here. So they don't want it. So they want to maintain at the highly concentrated so that it is equilibrium to the filtrate. Okay. So how does this happen? Filtrate normally is passed from the Bosman capsule to the proximal tubule, right? About the same osmolarity at the blood. Here, you can see the osmolarity is similar normally. After they already enter the descending part of Duke of Hinder, the osmolarity starting to increase. Because water is already reabsorbed. Why? Because water is already absorbed, meaning the concentrate solute is increasing. So the osmolarity starting to increase. So diffusion of salt out of the tubule is maximum as the filtrate round the curve and enter the ascending limb which is permeable to salt but not water. Okay. So NaCl diffusing from the ascending limb help maintain a high osmolarity in the interstitial fluid of the vena medulla. And the counter current system of the loop or handle involve active transport and thus an expenditure of the energy. So your active transport of NaCl from the filtrate at the upper part of the loop or hinder maintain a high salt concentration in the interior of the kidney and enable the kidney to form concentrated urine. So you can imagine that after you go down, when they want to reach the ascending part, NaCl starting to be a job. Why? Because they need to maintain concentrated region at the medulla. If no, meaning that here you will be meaning that your filtrate will be highly concentrated at here. But I don't want. I need to balance multiple current system, balance so that the water the osmolarity pressure was starting to decrease in the filtrate. So in the descending and ascending vessel of the vasa vata, they carry blood in opposite direction through the kidney osmolarity gradient. So so as the descending vessel convey blood toward the inner medulla, water is lost from the blood and the NaCl gained by diffusion. These net flux are reversed and blood flow back toward the cortex in the ascending vessel of the vessel. So with water re-entering the blood and diffusing out lah. So this is actually more focused on here lah. Here, vasa vesta here. Because you see that when the blood is also passed through here. Blood is also pass through near the loop of hinder. So in the descending and ascending vessel of the vesta, they carry blood in opposite direction. Meaning that in outer V, they carry blood in downward, but the filtrate is upward, right? Upward, right? As the descending vessel convey the blood toward the medulla, water was lost from the blood. Because you need to know that in medulla region it is highly concentrated. You can imagine that there are actually blood vessels at here. So when at here, meaning that water is reabsorbed, water is diffused out from the blood and enter the medulla region. So these net flux are reversed as blood flow back toward the cortex in the ascending vessel of the blood vesta with water re-entering the blood and salt diffusing out. Meaning that after they already the blood flow until here, meaning the water starting to enter back because it is hypotonic to the blood vessel, right? So as a result, the transport of the sodium chloride ion out of the thick segment of the ascending limb, the filtrate is actually hypoosmotic to body fluid by the time it reach the distal tubule. So that when you you continue to active transport the salt so that it is hypoosmotic region. So these they are the counter current system lah. Because the active transport of the NaCl is to maintain the osmotic pressure of the outside. So next, the filtrate descend again toward the medulla as time in the collecting duct, which is permeable to water but not the salt. So in collecting duct normally is permeable to water, not salt anymore lah. So therefore, osmosis extract water from the filtrate as it pass through from cortex to medulla and encounter the interstitial flow of increasing osmolality. So that's why they want to become hypoosmotic, so that when the filtrate move along the collecting duct, more water is absorbed, so that is osmolality pressure of the filtrate will be increased. If you imagine that if the osmotic pressure is all along the high one, actually you cannot produce the concentrated urine. So this process concentrates salt, urea, and other solute in the filtrate. So some urea will pass out the lower portion of the collecting duct and contribute to the high intestinal osmolality of the inner medulla. So that's all about the juxta medulla nephron in the concentrated urine formation. Okay, so this one is very important. Now we go through the osmoregulation in human. Just now is just a general formation of the urine. So now when you want to talk about ADH hormone, so ADH hormone, the hormone name is antidiuretic hormone lah. Okay, so what is happening here is that it is the homeostasis process, and what is the stimulus change, which is a blood osmolality or water potential lah. We may say blood osmolality normally is solute concentration of the blood. So meaning that when your blood osmolality is very high, normally is you are drinking less water, you are doing exercising, or you are eating too salty food, all that. When your blood osmolality is very high, your water potential in the blood is reduced. This is a stimulus, and this you will stimulate the osmoreceptor in the hypothalamus. And it's stimulated and release it is detected and stimulated, and this hypothalamus will secrete antidiuretic hormone ADH from the posterior pituitary gland. Gland. So meaning that hypothalamus will secrete the ADH, and this ADH actually is stored at the posterior pituitary gland. Okay, then it will release out from the posterior pituitary gland to the distal tubule and collecting duct. Collecting duct. So what happening is that it will increase the reabsorption of the water in collecting duct and distal tubule. So ADH will increase the permeability of distal convoluted tubule and collecting duct to water molecule. A aquaporin water channel will fuse to plasma membrane of the kidney tubule. So when the E is more permeable to the water, so it will increase water absorption. Water absorption to the interstitial fluid and then finally go to the blood. So the go to the blood. Cause the urine volume decrease, which is concentrated. So meaning that when the water is already reabsorbed, more water is reabsorbed from the collecting duct and distal tubule. Of course, your urine is highly concentrated. So that's why that when you not drinking too much water, you see that your urine is in yellow color, dark yellow color, especially when you are waking up. Because along the night you sleep, you're not drinking any water, right? So this is actually ADH play the role. They need to because your body require water. That you along you sleep, you do not consume any water, you do not drink any water. So they need water, so they reabsorb from the collecting duct and distal tubule. So, cause the urine volume decrease, we concentrated, you will produce highly concentrated urine. And for the blood osmolality, because they already receive the water, it will starting to decrease and return to the set point. So this is what you happening in the ADH lah. So here lah. So this is about the whether water potential of blood decrease, which blood osmolality is high. When your blood osmolality is high, so ADH hormone is produced. But however, when your blood osmolality is low, which the water potential is high, it also will be detected by osmotic receptor, but it will not release ADH. Will inhibit the releasing of ADH, or less ADH is released, so that it will not permeable. Your collecting duct and distal tubule will not highly permeable to the water, so water reabsorption in the distal collecting tubule and collecting duct decrease and then it will return to the set point. Okay. So this is just a response of how the ADH respond to the epithelial cell of the collecting duct in and distal convoluted tubule, and this diagram taken from Campbell. So ADH is non-steroid hormone. So of course the pathway is second messenger pathway lah, right? So it will activate protein kinase A, and this protein kinase A will cause the vesicle E with aquaporin to fuse with the plasma membrane of the epithelial cell, and your aquaporin which is a water channel present on the plasma membrane enhance the water reabsorption from the filtrate and reabsorb more water from the collecting duct and the distal convoluted tubule. So this is just a sample only. Okay, next we will go through the endotherm role in osmoregulation. Okay, so this is more spacious gate compared to ADH, because you need to know that ADH only respond to the blood osmolality only. They cannot respond when the both salt and body fluid are lost at the same time. So meaning that they cannot respond to the blood pressure and blood volume at the same time. So when your blood pressure or your blood volume drop, they will trigger the system what we call as RAAS, which is the renin angiotensin aldosterone system. So it is not only including aldosterone, we also including the liver system one. Not just like the ADH or got blood pressure volume drop, then it directly trigger aerosol release. No, they require more complicated response. So RS response would respond in the decrease in blood volume or blood pressure and increase of blood osmolality with ADH. So actually you can say that it is simultaneously lah. If the blood osmolality also increase ADH and aldosterone also can be able to secrete at the same time. But aldosterone more focusing on the blood volume and blood pressure drop. When the blood volume and blood pressure is decreased. So RIAS involved in these are JGA, juxtagomular apparatus here. E is normally located at the distal tubule. So it's a specialized tissue around the afferent arteriole. So when the blood pressure or blood volume decrease, the JGA, this juxtagomular apparatus, was starting to release renin. This is one type of the enzyme. So this enzyme, what happened is that you can see this diagram more easily. This decrease in blood pressure, it was starting to cause the JGA secret renin enzyme. So this renin enzyme will cause will catalyze angiotensinogen into angiotensin 1. Okay, what is angiotensinogen? Okay. So angiotensinogen actually is coming from the liver. The liver will secrete the plasma protein called angiotensinogen at the same time. So when it has the renin, it will catalyze the angiotensinogen into angiotensin 1. So when the angiotensin 1 is increased in concentration, it will cause the stimulate lung to secrete angiotensin converting enzyme, which is ACE, to the enzyme catalyze into angiotensin 1 into angiotensin 2. So meaning that renin catalyze angiotensinogen into angiotensin 1. Once angiotensin increase in certain amount, it will stimulate lung to secrete ACE. So this ACE will catalyze angiotensin 1 into angiotensin 2. Okay. Next, this angiotensin 2 will stimulate the adrenal gland starting to release secrete the aldosterone. So aldosterone is secreted by adrenal gland. So how to stimulate adrenal gland to secrete aldosterone is due to the angiotensin 2 concentration. Okay, once the aldosterone is already secreted in kidney, it will increase the sodium ion and water reabsorption. This is very important. Not only water absorption, but also sodium ion, and increase the secretion of potassium ion and hydrogen ion into urine. So if happening at the same time, first period is reabsorption, water and sodium ion is reabsorb because the blood pressure is already very low, your blood volume is very low, so they need water and salt. So at the same time, you don't need you need to balance the electrical charge. So potassium ion and hydrogen ion will secrete into urine. So it will increase your blood volume to the normal. At the same time, angiotensin 2 also will cause vasoconstriction of the arterioles. Is the blood capillaries. So this will increase the blood pressure and decreasing the blood flow to the capillary in the kidney. As a result, your blood pressure and blood volume will increase to the set point. So normally RAAS pathway will normally is cooperate with the ADH, but the response is different. ADH only respond to blood osmolality. Aldosterone will respond to both blood volume or blood pressure. Next, we will proceed to osmoregulation in plant, which is the final topic in this chapter. Okay. However, in this topic, I think it's very cold topic. Not say cold topic, boring topic, because it is just read and memorize. There are no such big mechanism like the previous one, but mostly maybe you will not read it, but I need to tell you that past year came out many times, especially in essay. Many times. Always repeat this kind of question. So it is actually very, you can say bonus to you lah if you read very well lah. Because it just memorize only. So because in the this chapter, I will refer to the syllabus. There are one topic is about the role of stomata and acetic acid in regulation of water loss by transpiration during water stress. So this topic actually I already uh explained in the first chapter lah. So you can, if you want to know more about this topic, you can refer back to the first chapter lah. Gases exchange, because at that chapter I think it's more easier to you lah. To understand, because at that chapter you already mentioned about gases exchange in stomata, causing of stomata. So directly you use this, explain it lah, more easy. Don't need to separate. If you separate, you will be confusing. Yeah. Okay. So in this topic, there are two type of plant that is very special, that can able to tolerate regulate their water load, osmoregulation in the extreme environment. First one is xerophyte, and then the next one is halophyte. We only learn these two only. There are also another two, mesophyte and also hydrophyte. Hydrophyte. Mesophyte is like a normal plant. Hydrophyte is the plant that survive in water, like water lily. But that one is not in the syllabus. In the syllabus, is only xerophyte and halophyte only. So xerophyte, if you see the diagram, cactus. Most example is the cactus. So of course you already know lah. Cactus is survive at the desert. They don't need much of water one. Meaning that this is the plant that normally survive at very hot, dry climate or live in the area that is very short supply in water. Uh, so yeah, so like desert lah. So one of the key example is cactus lah. So yeah, these three is the genus name, genus and species name. You can write it if the exam coming out want you to give an example of this. Okay, so you need to know what is the morphological adaptation and physiological adaptation of these two plants. Normally they come out this only. First of all, I will explain about the xerophyte stomata. So this is a morphological adaptation lah. So stomata is very important in plant, because it play the role to control the water loss in plant. If you imagine xerophyte, it is survive at the very high temperature part and low water condition, like desert, 60 degrees Celsius. Can you imagine if normal situation, like a normal plant survive at there, meaning that during the day, the stomata is open. Once it open, what happening? Water loss, and the plant will die. And because at there, no water, water supply is very short. So it's very limited. So they cannot get the water, then the plant will die. But how the cactus still can survive at there? So their stomata is different compared to the normal plant. So they have a normal or sunken stomata. So their stomata is sunken structure on their leaf. And the sunken stomata enable to reduce the surface area of leaf. So resulting a low rate of transpiration. So this is a spatial sunken stomata. And then the sunken stomata can trap moist air around the atmosphere. So this is the normal plant cannot do one. Normal plant cannot trap the moist air one, but sunken stomata can. And of course, the stomata is closed during the day and open during the night, like the CAMP plant. CAM plant, so not CAMP. CAMP is a short form. CAM plant, that you already learned in Sem 1 last chapter, right? CAM plant. Okay. So, and then the stomata are recessed in cavities called crypts. You can see this diagram. You can see the stomata is located at the upper part one. Upper part. But at here they are the cavities we call as crypt here. Here, sorry, here, crypts. And then at here we have many hairs. We have many hairs at the surrounding of the cells. So this is all the white color here is we call as quick. This is a cavities. So this adaptation reduce the rate of transpiration by protecting the stomata from very hot, driving environment. Right? Because it is located at the very, you can see that it is located at the very up region. So meaning that meaning that when in during hot condition, it is more hot condition, the heat is not easily transmitted from here into to the stomata, so that it can uh reduce the rate of transpiration. Uh, and also there are also the trichome. You can see here the hair structure like this. All these, you can see here is this is the hair like a hair structure minimize transpiration by breaking up the flow of air, allowing the chamber of the quick to have the higher humidity than the surrounding atmosphere. So it maintain the high humidity area. So that the high humidity area, so that the water transpiration from the mesophyll part not easily coming out through the stomata. So this is a spatial adaptation of the stomata in xerophyte. Okay, apart from the stomata, there are also other parts, including their roots. Their roots are very long, so that it can reach the underground water sources. And also the stem of the stomata is fleshy. Fleshy meaning that they can store water at the stem. Also their plant surface area covered with tiny hair to reduce airflow. Uh, uh, and the leaf are less amount, so that water loss is can be prevented. And also the leaf are succulent, and the leaf are able to roll up, and the photosynthesis is CNM metabolism lah. And cactus has shallow and adhesive root system. And this is a physiological adaptation. So including FCC acid, low root cell have very low water potential, high osmotic concentration, all this. Okay, next is halophyte. So halophyte is plants are adapted at very high salt concentration area. So one of the example is mangrove plant. Because mangrove plant. Okay. So this is adaptation. So normally they have the they are have thick layer of cuticle to reduce the rate of transpiration. Succulently store of water. Stomata is little, and then have tiny hair of the leaf surface, and then the root are arranged in Synatopore, allow oxygen to enter, and then leaf have the hydrotoid to remove excess salt, has sunken stomata similar to the xerophyte, and have both root and bulbous root for encourage and support in moody soil. And for the physiological is carried out by priority to increase chance of survival, secrete more FCC acid, secrete excess salt through hydrotoid, and actively accumulate salt into the plant, resulting the low water potential, allow water from the sea to diffuse out in the root xylem vessel by osmosis. High dissolved salt content in the root make it more harder, high transpiration pool, to small suction, high rate of transpiration. So that's all for this chapter. So thanks for those who are listening from beginning until end, and thanks for your supporting. Hope this video can provide helpful explanation to you guys about this chapter 12, homeostasis. I know this is very long topic, for big topic, and there are many new things that you need to learn. So hopefully based on this video you can understanding well. So if you have any question, you can ask in the chat box or join my Telegram group. So that's all for this video, and thanks for watching, and bye.